I want to welcome everyone. We're live from Asilomar now for the second time. And the first time, of course, we walked around and took nice pictures and didn't have and that was the meeting we all missed Asilomar for the ENC. Before starting, I'd like to introduce, I am Dave Rice, if I haven't said my name before, and Dan Iverson, and Krishnamurthy, and along with John Webb, we got together in 2016 to make a reality out of John's wish to have to found a meeting group. And so John came out to the West Coast and he invited us all to the Fairmont Hotel in San Jose. Also, I'd like to thank Eric, who is doing all of running our technical support, and Jake Hammond, who is helping him. If you're online, they have an incredible setup back there to both broadcast speakers from in here and from online. And Shelley Hammond, who is outside right now, she planned all the logistics for this meeting. And so we really thank her for all that hard work. And of course, most of all, to all of you online and who have come for this meeting this morning here, thank you for making Ivan a successful group and with a lot of successful meetings. So, Eric, if you want to show John's slides, I will go through them quickly. And if anyone doesn't know, John Webb is the founder and CEO of MR Resources, which I would describe as the premier third-party source for NMR parts and service and full NMR systems. So anyway, these are some of the things that MR Resources does. They sell spectrometers, use spectrometers, and lease them. They sell magnets, install magnets, and take care of magnets for people. And of course, they install consoles and offer service contracts, consoles, probes. They offer service contracts, and I guess quenched magnet recovery. And of course, if you need to move your magnet, they offer magnet relocation services. So, and the other venture that John is in is he is the MR Resources, or his organization is the distributor for our new NMR company, Q1, which has arisen in the last few years. And Q1, of course, offers new systems and all the sales and support that that the other two vendors offer as well. So Don Bouchard, who is here, will talk about Q1 later in the meeting. So I guess that's the slides that I'm to talk about. And so some other things I'll mention, come to the suite here in the evenings, and we will be having the Ivan Hours, and we have a set of 3 speakers, Monday, Tuesday, Wednesday, who will be talking about different subjects. Ron Crouch, who I saw here, will talk about the stars on Monday night, which I'm really looking forward to, and I'll let you look at the brochures for the rest of them. Also, I mentioned, look forward to our announcement of the winner of this year's Founders Award, and I'll keep it a secret until noontime for John to make the announcement. And be sure to stay here for lunch. And during lunch, we will also play a video from an individual, what's his first name? I forgot his first name, but Kornbluth. He is a World International Helium consultant, and who finds helium for people in the midst of the crisis. And so he's going to talk a bit about the helium situation. That will be a video playing during lunchtime while we have lunch in here. And also John would have said, will have said, and I will too, that we're always looking for new people to take up a role in Ivan and to give roundtable workshops and to plan academic events that they think that people should see. And Ivan is willing to, to get in there and make those things happen. So, and from my point of view, I'd make invitations for people that are in the bio world and in the also in the solids world, which is becoming overlapped with the bio world, to help suggest to me possible roundtable workshops. So with that, let's get started. So I'd like to introduce Alexandre Poulhazan. Yeah, sorry, I'm. And he's from Stanford University recently, but from the University of Montreal, Quebec before. He is going to talk about bio and solids, and his talk type is In-situ solid-state NMR approaches to characterize algal polysaccharides. So why don't you get started?
Thank you for the introduction, and sorry for the technical problem. So today, yes, I will talk about what I did during my PhD in Montreal, so at the University of Quebec in Montreal, under the supervision of Isabelle Marcotte and Dror Warszawski. So first, we study microalgae. Then, because they are found everywhere, they are found in freshwater, in oceans, and they have many potential added values that we know now. And now we also know it has many potential added values. First, you can think about nutrition, also biofuels, even now therapeutics, and they are also good candidates for bioremediation since they are able to bioaccumulate heavy metal, for example. Then for research, algae are also interesting because they are a good model for photosynthesis, for also glycoproteins heterologous production, and also they are a good model to simulate the glycans from higher plants. Also, we have already available an important bank of mutants for algae with cell wall starch mutant I will talk about today. And for NMR, which is what I will talk about today, it's also interesting because you can label them, isotopically label them, for not that very expensive, because you don't need glucose, you need instead a sodium bicarbonate to carbon-13 label those cells. Then we study glycans of those organisms. First, because glycans are found in almost all types of organisms, they are associated with a tremendous variety of biological function. For example, you can find just pure polysaccharide, and they are usually associated with energy storage and cell architecture. You can think about starch or cellulose in plants, for example. But it can also be found decorating many other biomolecules. For example, it can be decorating lipids, giving in algae the galactolipids, or it can also be decorating proteins, giving the glycoproteins. And actually, glycosylation of protein, as you might know, is the main translational modification happening on proteins. So it's interesting to know how glycosylation are happening on proteins, that we already know it's happening through the O-glycosylation and N-glycosylation patterns. So to come back to how I produce my samples and how we produce label samples, in our case, it's quite simple. You just have agar plates where you have your cells, you resuspend them in a carbon-13 nitrogen-15 labeled media with sodium bicarbonate and ammonium chloride. Then you leave your cell grow for about 6 days, and then you reach the exponential phase. You can spin down your cells, and this is how you get your crude whole cell spin down in your tube. So from these cells, we can just study, I will talk about two different type of algae. First, Chlamydomonas lunardi, that we can extract the starch. That will be the first part of my talk. Then I will talk about another algae that we can just study as it is, just whole cell, and I will talk about assignment and quantification of whole cell glycans. Then I will switch back to an extract from Chlamydomonas lunardi algae, talking about the glycoproteins architecture into the cell wall of this algae. This overall to hopefully show you that solid-state NMR is a powerful tool to have more insight and very atomic scale details of even complex biological systems. So as you know, NMR gives atomic scale information. Unfortunately, just using 1D NMR, which is possible for even unlabeled samples, doesn't give very high resolution and can be limited even more for solid state NMR. So we need 2D experiments. So therefore, you usually need carbon-13 labeled, yes, but you will gain a lot in terms of resolution because you have a new axis to disperse your peak. And the experiment we use is called the inadequate experiment that you might know. And it actually allows not only to have a single quantum coherence, but instead we have a double quantum here on the y-axis. And instead of having the usual chemical shift, we have the sum of the two the chemical shift of the two correlated carbons in this case. So it allows us for glycans to go through the what we call the spin system there of a glycan. And here you can see the numbering I use through all the presentation. So from just starting from the carbon 1, we can go through all the glycan ring up to the carbon 6, because all those carbons are all linked chemically. So this experiment is very interesting because of this chemical dispersion along the y-axis, and we get rid of the diagonal that might be annoying when you are very close chemical shifts. Then you can also combine that with different magnetization at the beginning, direct pulse, cross-polarization, showing different types of dynamics and objects in your sample. But don't forget that, unfortunately, so far, you still need carbon-13 labeled, or at least MAS DNP that might improve a lot of sensitivity. So first, as I said, I will talk about the starch of chlamydomonas, which is just a way for me to show you how I use the inadequate experiment and this chemical shift dispersion. First, you know starch is a main energy storage for plants. It's made of glucose units. They can be linked through two different kinds of chemical links, the alpha-1,4 that will create the long linear amylose, but you can also have a more branched structure called the amylopectin with alpha-1,6 linkages there. From these two different components, amylose and amylopectin, you can also have a diversity of structure that can be detected directly using electron microscopy, but you will, in those cases, you will always have an interplay of amorphous and crystalline regions. And talking about those crystalline regions, there are double-stranded helices like that, and the way they will be all together will define two types of starch, called the A-type and B-type starch. And as you can see, there are just very small differences between those. We can expect very small differences in terms of chemical shift between those two types of starch, and indeed, we just have one difference is on the carbon 1 chemical shift, where we'll have 3 peaks for A-type starch and only 2 peaks for B-type starch. As you will see just here, so in Chlamydomonas renardii, we have A-type starch. What we can do is that we can boil it and make it amorphous, and the way it will recrystallize as by itself will make a B-type starch called a retrograded B-type starch. And as you can see again, 3 peaks for carbon 1 and only 2 peaks for A-type and only 2 peaks for B-type starch. But again, 1D is insufficient in terms of resolution to assign all the peaks. So we went to the 2D inadequate experiment, and the paper we published in 2018 was the first one to report all the chemical shifts for both types of starch. We also noticed the same differences as the 1D, 3 peaks for A-type starch while 2 peaks for B-type starch, but the 2D also allowed us to see another difference, this time on the carbon 5, where we just have 2 peaks for B-type starch and only 1 peak for A-type starch. But I don't know if you noticed there, the flashing part in green is something that were not assigned before, because it's lower and it's overlapping with the carbon 6, carbon 5 of usual starch. But inadequate experiment allowed us to see that, okay, definitely we have a spin system there that is not assigned. And comparing it with the literature, we found out it was the non-reducing end group of the starch. As a polymer, you have an end of it, so the more ends you have, the more intense this spin system will be. So it means having an idea of this intensity will give us an idea of the chain length of the starch. So with that, we proved that indeed we can do that, comparing the aminopectin that is known to have short chains but very branched, that have therefore a much higher signal there, comparing it to the linear amylose that is a long linear polymer but with fewer non-reducing end groups, and it worked with the inadequate there. Few conclusions on the starch. Again, we assign all the carbons of both types of starch and identify the non-reducing end group. Again, we need this inadequate experiment, so usually carbon-13 labeling, and we also did some in-cell measurements, allowing us to have an idea of quantities directly in the cell of the starch and also following the type of starch directly in the cell without any modification of the sample. But as you might think, like starch is a big one, is a big carbohydrate, easy to identify and easy to see, but we have many other one glycans in the cell because starch is just one spin system in a whole cell sample, as you can see here. So we have many other peaks. So we wanted to develop a method to have not only starch, but all the glycans directly from the cell assigned and quantified using solid-state NMR and whole cell. So that's why I switched back to another algae, Parachlorella bergerinki, that has a different cell wall, different composition, and Chlamydomonas renarti. But here is just a zoom in on the glycan region of this whole cell spectra. Using a high magnetic field, 800 megahertz, and a high efficiency probe at the National Mag Lab in Florida, we managed to assign quite a lot of different spin systems in this sample using databases that are already available, such as the CCMRD database, and we assign the different glycans coming mostly from the cell wall, but also from galactolipids coming from algae membranes. We also compared those results with mass spectrometry that is able to also, again, just identify the units, but not the polysaccharide, and also quantify them. And our method has been proved to have a good match between solid-state NMR and mass spectrometry, proving that, indeed, solid-state NMR, if you have good resolution and you take care of your pulse sequence and relaxation, then you might have something quantitative. So just a quick, so that was the methodology we proposed in our paper. And we also, overall, more than a method, it was the first time we had a full assignment of most of the glycan of a single unicellular cell. But something you have to I have to remind you is that you have to take care and to take into account the T1 relaxation that will, of course, kill your signal, but not the same way for all the glycans. So you have to take that into account using usually 1D experiments. Then we also, in this paper, noticed not only assign the glycans, but we managed with other experiments to detect the contacts between different glycans in the from the cell wall with xylane, but also within the starch units, which has different spin system in it. So this methodology, which is kind of long to apply because you have many peaks and you have to consider many different chemical shifts, we wanted to apply it to, let's say, what most people would think an easier sample, but it still reveals some complexity. So that's why we'll switch back to an extract from Chlamydomonas renarti, but this time the cell wall of this algae. So the cell wall of Chlamydomonas renarti is already quite well known and has already reported many different layers, about 6 layers that are have also that kind of vesicle granule things in it. But already reported protocols managed to have part of the cell wall extracted using KH agent. In our case, it's sodium perchlorate, and we can simply reconstitute this cell wall using dialysis, leaving the cell not destroyed, but intact with just few layers of the cell wall left, which are known to be the non-KH agent soluble. But using solid-state NMR, what you can do is you can actually study both of all of those samples and make the sum of the part and just study one of them. But today I would just focus on the cell wall extract of this algae, which is known to be made of glycoproteins. The cell wall of Chlamydomonas renarti is really acting like a shell when we do whole cell experiments. Indeed, the cell can survive more than 24 hours in our solid-state NMR rotors spinning at 15 kilohertz without much of temperature control. So those are really survivors compared to like mammalian cells and that kind of thing because of their cell wall. So it has some mechanical properties interesting to understand and to study. This cell wall is a non-cellulosic cell wall compared to higher plants. Instead, it's made of glycoproteins. The main components that are reported are called the hydroxyproline-rich glycoproteins that are very large proteins, and they've been reported to be rich in arabinose and galactose units in terms of glycan decoration. So that's what we have in when we did our sample extraction compared to like previous literature. But we also found out that in our extract, because we did less, let's say, purification of just the big proteins, we also found out we had a lot of smaller proteins. You have even other ones, smaller molecular weight there. And those were actually rich in mannose units, which were unexpected and reported before. But still, we went further and study them against using the same methodology, the inadequate experiment, allowing assignment and quantification of not only glycan but also amino acids. I will also show to you other methods we use based on dipolar contacts between carbons and what is called the PDSD, as you might know, experiment, where you see the contacts between carbons, but we'll more focus on the transfer efficiency and decay of this type of experiment. Then I will also show you difference of hydration that can be probed using solid-state NMR experiment. And finally, I will take advantages of the MAS-DNP high sensitivity to detect the contact between amino acids and glycans. And this, at the end of this part, will lead to a model of the cell wall of this algae. So first, as before, inadequate experiment, a zoom in on the glycan region. We can assign quite a lot of different glycans, having good agreement between the mass spectrometry and solid-state NMR quantification. But as you might know, since I said it was glycoproteins, we don't have only glycans, we have also amino acids. So we apply exactly the same kind of approach on this same inadequate experiment, assigning quite a lot of different amino acids. So it's a bit small, but the idea is that we have many peaks. It takes months. I had months during my PhD. So I did that, assign, quantify, and again, good agreement between solid-state NMR and mass spectrometry. And what solid-state NMR can bring is that we are in native samples, so we can even have an idea of the secondary structure of our proteins, for example. So overall, it's about more than 500 peaks, unique peaks assigned for about 30 different glycans and 80 amino acids, different like conformation of amino acid units. So this is the first update of our model. Before we knew the HRGPs, but now in our current model, we know there are also lower molecular weight proteins rich in mannose. New update in terms of dynamic for this model is that first we measure the usual T1 relaxation of carbon-13, showing something very homogeneous. So that what we can say is overall our cell wall is quite dynamic at the nanosecond timescale. But we also use another experiment called the well-known cross-polarization that can probe different types of relaxation and build up. And comparing the carbohydrates with the amino acid signal, we prove that carbohydrates have a faster transfer efficiency, but more importantly, have a slower decay, proving that compared to amino acids, glycans are more rigid, but on the millisecond timescale. So this is a new update showing that in our model, in our system, we have rigid glycans compared to more dynamic amino acids. Then something else we can probe using solid-state NMR, and something that is maybe a bit more unusual, is to probe the hydration levels in our sample. Indeed, when we study the cell wall, we don't just put the powder, we hydrate this powder before putting it in our rotor. And we can use first the usual DAR experiment, where you see dipolar connection between different carbons as a single quantum coherence here. But what we can do before that, we can add a block to filter to T2 using the T2 relaxation of protons. We can just have the proton from water being excited. So therefore, we'll just have water exciting the carbons surrounding it before detecting it. And this is the resulting spectra we get here. And as you can see, most of the glycans, they survive this kind of filter, but all of the amino acid, they kind of disappeared, showing that indeed we have different discrepancies between amino acid and glycans in terms of hydration. But more than the spectra, we can have some quantification using them and having this kind of bar diagram showing that indeed amino acids are less hydrated than glycans. But we also prove that here, for example, you can see here, it's the hydroxyproline peaks, and proving that glycosylated amino acids have an intermediate behavior between the two, which is a new update again of our model, having the more hydrated glycans compared to the less hydrated amino acids, and the glycosylated amino acids having an intermediate behavior in terms of hydration. Then we wanted to detect, first we noticed that using the inadequate experiment where the hydroxyproline were expected, we didn't detect much of them. Even if we were at low abundance, we were surprising by the low abundance, like on the actual spectra. And we noticed that it might be some dynamics issue. So that's why we take the advantages of the low temperature of MAS-DNP without the sensitivity enhancement. So with the microwave turned off, so just low temperature, 100 Kelvin, we noticed that indeed We lose in terms of resolution, yes, but hydroxyproline are back. Just a quick note to show that inadequate, some dynamics can be a problem for inadequate experiment, and hydroxyproline seems to be in the range of those dynamics that are hard to detect for inadequate experiment. But what was the main idea here is to use MAS-DNP, which is a way to improve a lot our signal. And just very briefly how it works is that you have in your sample radicals, which mean unpaired electron, that you can excite using not radio frequencies but microwave. And then you can transfer this huge magnetization from proton then to carbon. Overall, in our case and in most samples, you can have a signal enhancement of more than about like 50 times signal enhancement, meaning that even unlabeled samples can be probed with this kind of approach, even if you lose in terms of resolution because of the low temperatures. But still, we managed to finally detect contacts between amino acid and glycans, and specific contacts between amino acid and glycans, showing that hydroxyproline were more prone to have contacts with actually it should be written arabinose and galactose and even mannose because there are so much of them. But we also prove some contacts between different glycans too in our model. So this was again a new update of our model with different contacts between amino acid and glycans. Some conclusion on this model is that overall the cell wall, so we managed to have an assignment and quantification of both amino acid and glycans. We proved that overall the cell wall is quite dynamic and hydrated. We also detected the HRGP that were already reported, but we also detected other smaller proteins that are much more abundant in this sample and that are rich in mannose components. Even if it's overall very dynamic and hydrated, we still managed to see some differences between the two species, proving that glycans were much more mobile compared to amino acid parts. And we also proved that the glycans region were much more hydrated compared to the amino acids. We also detected using the high sensitivity MAS-DNP experiment glycan-glycan contacts, but more importantly, some glycan amino acid contacts that are from intramolecular but also intermolecular contacts. Overall conclusion of this presentation is that I hope I showed to you that bio-solid-state NMR is something that is possible. You need, for like, let's say, fancy experiment, you need carbon-13 label, but even non-label samples can be studied. Using whole cell, we can already have some information, so no need to any extraction. It's a native cells. You can just run and have an idea of the bigger object in your sample. Then I also proved that some extracts are interesting to study and possible to study using solid-state NMR. And I talked just this morning about starch and cell wall. And overall, it just led to a better understanding of the composition of an architecture of microbiology components. Just few ideas for the perspective of our current work. We are currently studying and have better idea of other parts of the cell. For example, lipid droplets that can be studied by NMR, yes, but usually also more by confocal imaging. We have also the idea to use those organisms to probe the bioremediation capacities of those organisms by following where, for example, heavy metals are going to the cell. And we might use the paramagnetic effect or properties of those elements because it would just turn off some signal from the cell. So now we've assigned quite a lot of molecules. We can compare different samples and have an idea of what signal decrease and then come back to what was actually affected and where the heavy metals bioaccumulate. Overall, we can have an idea of all the stresses. Now we have assigned quite a lot of different molecules in the cell. For example, again, heavy metals, but also temperature, pH, that are important parameters to take into account now for ocean and global warming conditions. And we also can probe and and follow what are the effects of bioengineering. For instance, you can design cells to make more lipids for biofuels. So we can just spin down the cells and follow the lipids contents directly and just live kind of thing. So as I said, other parts are also explored with the lipids or, for instance, the flagella that are interesting for other karyotic cell systems. So with that, I would like to thank my the people from the UQAM University, like Professor Isabelle Marcotte and André Warszawski, my co-supervisor, but also Alexandre Arnold, our NMR manager, that all three of them, they did a lot to help me during my PhD and all the work I just presented today. And I also did a 6 months internship with Professor Liwang, that is now not anymore at the LSU, but now in Michigan State University, where I did, and I've been in collaboration also with the National Mag Lab, so that's where I did the wholesale experiment kind of thing, and assignment. So I also thank people at the National Mag Lab, Ivan Heng for the high field, and Frederic Mentic-Viguier for the MES DNP. And I also thank now Professor Segelski, where I'm doing my postdoc just now. I just arrived a few months ago, but hopefully next year I will present stuff from Stanford. And thank you for your attention. And if you have questions.
So thank you for a great talk. Are there questions? Ron?
Listen, the inadequates were very, very interesting. I was curious if you see strong coupling effects in the inadequate in the solid state from two close carbons. Have you observed that?
Well, actually, what we noticed, it's a very interesting question. So we noticed that indeed using high resolution, we noticed a splitting on the carbon because of the surrounding carbons, and so we were not sure actually where it came from. Our hypothesis is that it was J coupling coming. The splitting was coming from the J coupling, so it's from the wholesale experiment. So we need high resolution to see this coupling. You see, on the starch resolution is not sufficient. It was a 400 megahertz, or even maybe 600. This one, but here using the high efficiency probe, we notice that here you see almost all the peaks are kind of splitted. And our hypothesis, so yes, to answer your question, yes, we saw that. And our hypothesis was that it's coming from the J coupling, but it was much higher compared to what is observed in solution NMR. But J coupling should be the same between two linked carbons. So that's why I was surprised. So we were, instead of having 40 hertz, we are up to like 60, 70 hertz splitting, which was quite surprising. But that's what we did that's what we said in our papers, that it's probably coming from the J coupling that can be detected.
Yeah, in the inadequate, Bax published back in, good heavens, maybe 1981, there was a suggestion that you could use 3/4J optimization instead of 1/4J optimization, and the strong coupling stuff becomes much more intense.
Ah, okay, we didn't
That
try
was it. Very, very interesting talk. Thank you.
Thank you for the question.
Another question? Oh, go ahead.
Very nice talk, thank you. When you were measuring chain lengths of your oligosaccharides, did you do that from your 1D or from your 2D?
2D.
Cross-peak intensities?
You can actually, we did both, and it gives similar results. So we can use, since we have, but it's just using 1D, it's just a very small shoulder on this part of the spectra. So you will need like high resolution to know exactly what you have. But using this inadequate experiment, we didn't have any standard to have an idea of how precise we were, but we can just take this peak that is very alone and integrate it and compare it with this one, which is also very separated from the other one. And using this 2D, that's how we had an idea of how many times longer this chain was compared to the other one. But we didn't say it was like 50 units because we had no standard.
Yep.
But they are probably available, but for those kind of long objects, we can expect that this process quantification might not work perfectly, but it gives you an idea of this length of your polymer.
Great, thank you.
More questions? So I have a question. Could you, the DNP setup that you used, could you describe that in more detail?
Yeah, sure. So that's the setup they have at the MagLab lab. And so it's, I didn't want to go into much detail to make it not too long because it's a morning.
Yeah.
Morning, so just after that. Okay. So just briefly, that it's you will need a gyrotron to add to your usual NMR to be able to create or to to make your microwave, you need then on their setup they have a semi-optic bench, so they can kind of filter what the microwave gyrotron is making. Then you have a waveguide, so then you can just irradiate your sample with those microwave, and in your sample, which is in a sapphire rotor to be microwave transparent. You can also use diamond if you're like very fancy, but sapphire is already expensive and works. Yeah. And even the zirconia works, if thin wall. So anyway, so you have your radical in your sample mixed with your molecules. You have some cryoprotectant because MASDNP needs very low temperature, 100 Kelvin, so everything is frozen. But we don't want ice crystal, so we put glycerol. You can even do wholesale, and they might survive to it because you have cryoprotectant. So you have your radicals. So microwave, you will just have your, you will just excite your electron, your unpaired electron from this radical, and then you can transfer it to proton. Like that's a huge magnetization. Then you transfer it, and you have different. I know this here. I have rough idea of the theory of it, but I know it's very complex. Yeah. And you have different effects that can transfer it. Right. And I know in our case, the main effect is the cross effect that can transfer this magnetization from the radical to the proton, and then is the usual experiment you can have, like proton to carbon and then coherence between different carbons.
Okay. And you get 50 times?
Yeah. Yeah, that's for real.
Quite amazing.
Yeah, so you can
for
just
these
have
kinds of experiments.
Yeah, and even with in theory, you can go up to like, what's the theory of it? It's like, is it 200 or even more than that? But in like with actual sample, it's never like that. But with new design of radicals that Frédéric Maintig-Viguier is also working on, we had even like 60 times enhancement on wholesale and extracted sample. So it's life-changing for people that can't label their sample.
Right. Okay. So any more questions? If not, let's move on to our next segment here. Thank you very much, Alexandre.
Thank you.
We're now going to be giving out our first prize of the morning, and that will be done through a lottery in which everyone here and online is represented in the pool. And Dan is going, as last year, Dan is going to choose the winner with a fancy program run from VNMRJ. And so I didn't mention the prize is a $50 gift card. So Dan, take it away.
Here we go. And the winner has to be present, but there it is.
Oh, there we go. Yeah, da-da-da-da-da-da. Yeah. William Hiscox, are you out there? Just speak up from Zoom if you're out there. He's doing the meeting. Okay. He's logged into the Uh-oh, going to get coffee. He'll miss the prize. Okay, how are we doing there for time? He's over. Okay, try again.
Try again.
Dada. Hey! Congratulations, George. Do you have the camera on him? Or that's all right, that's too complicated. So, Shelley, our prize giver, is okay, great. So while that's going on, I'll move on to the next speaker. And our next speaker comes from online and is a good friend of mine at UC Merced, Andy Liwang. Okay, so his talk is using protein NMR to study a circadian clock. And
yeah, hi, my name is Andy Liwang. Thank you for inviting me, Dave, and thanks to Eric as well. It's great to be here. I'm a biochemist at the University of California, Merced, and Dave and I have been working together for many years, and it's great to have him as a colleague. So this is an aerial photo of UC Merced, and we're close to Yosemite National Park. So we're up against the Sierra Nevada foothills. So if you love to hike and camp, this is a great place. And we're about 2 hours away from Asilomar. I wish I could be there in person, but yesterday I just flew in from another conference. So it's been a little hectic. So thank you for letting me present remotely. The last talk was really, really cool. Okay, so we use NMR to study circadian clock. And why would we want to do that? Well, it turns out that circadian clocks drive metabolic rhythms in anticipation of day and night. So that's really the importance of the circadian clock, is that most life forms have evolved under the rising and setting of the sun. And so due to the dramatic differences between day and night, it's not surprising that organisms have adapted to that by evolving these clocks in anticipation of sunrise and sunset. And you find circadian clocks in mammals, plants, fungi, insects, and bacteria. Just so it's a fundamental biological system. In fact, in 2017, the Nobel Prize was awarded to these scientists for their pioneering work on the genetics of the circadian clock of the fruit fly. Even Charles Darwin was fascinated by the circadian movements of the leaves in plants. And so you can download his book online. I think it's free. And he has numerous drawings in there of plants during the daytime when the leaves are up and out trying to collect all the photons from the sun, and then at night when they droop. Now, my lab, in trying to understand the mechanism of circadian clocks, really decided to choose the clock of cyanobacteria because they have the simplest known clock. Just to show you a little video of the circadian rhythms in cyanobacteria, and this video is taken by my collaborator Susan Golden at UC San Diego. She streaked out cyanobacteria in a moon shape on a plate, and then also in a sun shape, and then she synchronized them 12 hours apart. And these bacteria have been genetically altered so that one of the clock-controlled promoters drives expression of luciferase, and so you can record the bioluminescence from these cells. So let's just watch. And so these little guys have a circadian clock. And as you know, bacterial systems are usually much simpler than eukaryotic systems. That's why our standard undergraduate biochemistry textbooks, a lot of the information in there were derived from studies on bacteria. In 1998, these three scientists discovered the three core clock genes of cyanobacteria, and they're called Chi-A, Chi-B, and Chi-C. So this was a big breakthrough because now that the genes were discovered, people could clone these genes and then express them recombinantly in E. coli and then study the biochemistry of these proteins. And this is approximately when I joined the field as an assistant professor. Then in 2005, Tako Kondo's lab also made a huge breakthrough, which is that they discovered that if you mix recombinant forms of Chi-A, Chi-B, and Chi-C together in a test tube with some ATP, that just these three proteins will set up an autonomous macroscopic rhythm of Chi-C phosphorylation and dephosphorylation as seen as this gel. And this phosphorylation pattern of Chi-C recapitulates the phosphorylation rhythm of Chi-C found in live cyanobacteria. So then what this showed was that now we could study this clock in vitro using biochemical techniques, biophysical techniques under highly defined conditions. And as you know, trying to study the mechanism of anything, any protein or protein system in live cells presents several daunting challenges. So this breakthrough was big for biochemists. Now, around the same time as Kondo's discovery, the clock proteins were all crystallized. So Chi-A crystallizes as a domain-swapped homodimer, Chi-B crystallizes as a homotetramer, and Chi-C crystallizes as a homohexamer. And this here, the C1 and C2 here, that's one subunit of Chi-C. The C1 is the N-terminal domain, the C2 is the C-terminal domain, and you can see they're connected by this green linker. And the C2 domains self-associate to form a ring. The C1 domains self-associate to form a ring. And you can see these ATP molecules colored in red bound at the interfaces between the subunits. And Chi-C autophosphorylates and autodephosphorylates at these two residues colored in cyan. And one of them is a serine residue, and one of them is a threonine residue. And we'll talk more about them coming up. And these purple things that are tucked into this pore on the top of the C2 ring, these are called A loops. And then there's a C-terminal extensions that come off of these A loops, but you can't see them. They're not resolved in the crystal structure. A former student of mine found a convenient way to watch the clock function. And what Joel Heisler did, my former student, is he tagged Chi-B with a fluorophore, and instead of taking time points by hand and then running them on a gel like Tako Kondo did, which is extremely laborious, especially, you have to stay up like 3 or 4 nights straight to take those time points. Now with the fluorescent probe, we could just watch the oscillation in real time in a plate reader. And you can see this thing just keeps running and running. And at some point we just stopped the measurements. But I still kind of regret we didn't let it keep running, because I would like to see how long this thing would go. And remember, this is just 3 proteins in a aqueous solution with some ATP. Okay, so in the early days, we really want to know, since there's these 3 proteins, Chi-A, Chi-B, and Chi-C, this is in the early days, we want to know, like, what does Chi-A do in terms of Chi-C autophosphorylation and autodephosphorylation, and what does Chi-B do? So Young-Ik Kim, a former student of mine, what he did was he looked at Chi-C phosphorylation, shown here on the y-axis, over time. And if he looked at Chi-C by itself or with just Chi-B, he found that Chi-C, just kind of stays at this hypo phosphorylation level, so not very much phosphorylated. But if he mixes Chi-A with Chi-C, what he found is that Chi-C starts to autophosphorylate, and then it reaches this hyper level of phosphorylation. This gel is just from Tako Kondo's paper showing you that Chi-C autophosphorylates and autodephosphorylates with a circadian rhythm when all 3 proteins are together. Okay, now when Young-Ik then mixed Chi-B in with the Chi-A, Chi-C mixture, initially he sees that, as shown in these pink squares, he sees that the trajectory of Chi-C phosphorylation matches that trajectory when it's just Chi-C and Chi-A. But then at some point around midday, the trajectories diverge, and the sample with Chi-B in it, you can see that Chi-C starts to autodephosphorylate. And so this shows that Chi-B inhibits Chi-A's ability to stimulate Chi-C, but of course at this point in time we didn't know how. So we really want to know, how does Chi-A stimulate autophosphorylation of Chi-C? How does Chi-B block Chi-A? And why does Chi-B wait until around midday here? Young-ik first wanted to know how does Chi-A stimulate Chi-C autophosphorylation. And so we solved an NMR structure of Chi-A bound to a peptide that represented the A loops of Chi-C and the C-terminal extension shown here in gold that's not resolved in the crystal structure. And when he compared the NMR structure here with the position of the A loops here, he realized that the A loops would have to come out of this pore, have to come out in order for Chi to bind the A loops. And so what he did was he did a series of biochemical experiments where he destabilized the A loops so that in this position so that they would have to come out or want to come out. And he noticed that those mutants of Chi-C autophosphorylated even in the absence of Chi-A. And then when he deleted the A loops, when he just deleted them completely to mimic their exposed state, he found that Chi-C was hyperphosphorylated even in the absence of Chi-A. So then what he realized is that when the A loops are exposed, Chi-C autophosphorylates. When these A loops are buried, like shown in the crystal structure, Chi-C autodephosphorylates. So these A loops are really a switch that turn Chi-C on and off between autophosphorylation and autodephosphorylation. So Chi-A works by stabilizing the exposed position of these A loops. And because this is an NMR talk, and I haven't mentioned NMR yet, this structure here was solved by NMR. All right. So around 2011, this model of the clock came out. And so here is a cartoon of Chi-C. Here's the C2 ring up here. Here's the C1 ring down here. The A loops are shown as these little strands. And this is in the morning. And in the morning, Chi-C is not phosphorylated. And the two residues I showed you earlier that get phosphorylated and dephosphorylated that reside in these subunits of the C2 ring, the serine and threonine shown here, they're not phosphorylated. But Chi-A, as you now know, binds to those A loops in the C-terminal extensions and stabilizes those. Okay. I hear an echo. Okay, all fixed? All right. So this is in the morning, the unphosphorylated Chi-C state. And in the presence of Chi-A, Chi binds to those A loops and C-terminal extensions, stabilizing them in an exposed state. So then Chi-C starts to autophosphorylate. And other labs found out that the first residues to autophosphorylate are the threonine residues in the C2 ring. So each of these subunits, each of these domains in the C2 domains has a threonine residue that autophosphorylates. And here you see Chi-B, and it's drawn as a homo tetramer, and it's just sitting out, in solution, not doing anything. And this happens right now is about midday. And then under continued stimulation of Chi-A binding to those A loops, then the second residue to autophosphorylate is the serine. Once the serine residue autophosphorylates, people discovered that that's when Chi-B binds to Chi-C. And here it's showing that Chi-B is binding to the C2 ring near the sites of phosphorylation, and that somehow inhibits Chi-A. This happens in the evening. So now the sun is setting. And now we're going to enter nighttime. So now with Chi-B inhibiting Chi-A, Chi-C starts to auto-dephosphorylate. And it auto-dephosphorylates first on the threonine residue. And you can see that Chi-B is drawn here, somehow inhibiting Chi-A. And so with Chi-A unable to stimulate Chi-C autophosphorylation, Chi-C continues to auto-dephosphorylate throughout the night until it completely auto-dephosphorylates by morning. And once the serine auto-dephosphorylates, Chi-B can no longer bind. So Chi-B falls off and it releases Chi-A so that Chi-A can start a brand new cycle of Chi-C autophosphorylation. So this was the understanding in 2011. And people thought pretty much that the circadian clock of cyanobacteria was figured out. But some questions still remained was, for example, what regulates the Chi-BC interaction? Why does Chi-B interact with Chi-C only when the serine is phosphorylated? That was unknown. And then what is the role of the C1 ring? And you can see the C1 ring is sitting in each of these little panels, and it's not doing anything, apparently. All the action is happening on the C2 ring. Chi-B binds to the C2 ring, and then Chi-A is inhibited by Chi-B on the C2 ring. So we want to look into that a little more. So there's NMR coming up. Don't get too worried. This is important, though, for laying the groundwork. So Yonggong Chang, former postdoc in my lab, wanted to understand, what does phosphorylation really do to Chi-C? So he looked at isolated C2 domains of Chi-C and just ran it on a size exclusion column, and he found that it mostly, and this is fully dephosphorylated C2, and he found that it mostly comes off the column as a monomer, but a little bit comes off as a hexamer. This is the morning state of Chi-C. Then he wanted to use NMR to look at the full Chi-C, which is 360 kilodaltons. He methylated everything. It's quite expensive. You have to feed your E. coli deuterated glucose, grow them in D2O, and then feed it back precursors of the amino acids with methyl groups. In this case, we're labeling one of the methyl groups with isoleucine. And he's looking at signals coming exclusively from the C2 ring. And here you see very weak signals coming from the C2 ring. And, that kind of suggests that, well, maybe the C2 ring, it has these intermediate timescale motions that might reflect the breathing of the C2 ring. And that sort of makes sense because you can see that isolated C2 domains do not form stable hexamers. So this suggests that the C2 ring isn't really very stable as a hexamer, perhaps even in the context of full-length Chi-C. Then he looked at the midday state of Chi-C. So the midday state of Chi-C is when the threonine is phosphorylated. Now, to mimic threonine phosphorylation, he used a glutamate substitution. And here he sees that by size exclusion chromatography, isolated C2 domains run exclusively as monomers. So it seems like phosphorylation of the threonine destabilizes the C2 ring. And by NMR of full-length Chi-C, you can see the signals coming from the C2 ring are also, just like before, very weak. And then he wanted to look at the evening state of Chi-C. This is when the serine gets phosphorylated. And remember, when the serine gets phosphorylated, Chi-B will bind to Chi-C. So when he looked at the doubly phosphorylated state, and he used a double glutamate substitution to mimic phosphorylation of the serine and the threonine, he now saw that isolated C2 domains now run significantly more, as before, relative to the previous phosphorylation states, more as a hexamer. And then looking at the NMR signals from the C2 ring of Chi-C, he sees now nice tight signals coming off the C2 ring. And that suggested that, the C2 ring is tight or tighter than before. And then he looked at the late night state of Chi-C. The late night state is when it's just the serine is phosphorylated because the threonine has dephosphorylated already. And so if he looks at where just the serine is phosphorylated, so this is deep into the night, you can see that now the C2 ring is tight. It's running mostly as a hexamer, very little as a monomer. And then in the context of full-length Chi-C, you can see that the C2 ring has these nice tight signals. And when the C2 ring is tight like this, if you look at the A loops are forming these hydrogen bond electrostatic network, which helps stabilize them inside the ring. And this makes it harder for Chi-A to grab to those A loops because those A loops have to come out for Chi to grab them. And so when the ring is tight, Chi-A really can't stimulate Chi-C to autophosphorylate as well as when the ring is loose, which is when the threonine is phosphorylated. So you can see that the threonine, when it's phosphorylated, provides positive feedback to the phosphorylation of Chi-C by Chi-A. And then when the serine is phosphorylated, it provides negative feedback to the clock mechanism. So because this is an NMR talk, I'll just say that this explains why this phosphorylation order is in this direction and not in the opposite direction. So this is the clockwise phosphorylation direction. The anti-clockwise or counterclockwise direction would be having the serine phosphorylate first and the threonine phosphorylate second. And if you think about it, you can actually, just from this data, you can sort of explain why the clock phosphorylates this way instead of the opposite way. All right. Now let's move on to trying to understand how does Chi-B inhibit Chi-A. So remember, Chi-B inhibits Chi-A around midday. And so Yonggang looked back at the literature, and there's this low-res electron microscopy experiment where people looked at the Chi-C by itself and then with Chi-B. When they added Chi-B, they saw some extra density up here. And they said, oh, okay, so this must be where Chi-B binds on the C2 ring. Well, Yonggang was looking at this data. He didn't quite believe that the authors understood unambiguously which ring was which. So he sliced Chi-C in half and then looked to see which of those rings Chi-B bound to. So he added Chi-B to just the C2 ring, and he added Chi-B to just the C1 ring. And what he found was that Chi-B binds only to the C1 domains or ring and not the C2. So here you see just a simple HSQC of N15 labeled Chi-B plus minus the C1 domains of Chi-C, and he saw this massive change in the HSQC, which is not what you normally see when you add a binding partner. You usually just see a handful of peaks move, and those are usually near the binding interface. But here, almost every peak has shifted. We didn't understand the significance of this until a little bit later. But when he repeated this with adding C2, he didn't see any change in the C2. He didn't see any change in the Chi-B spectrum. So what this showed, which is in contrast to what people thought, is that Chi-B actually binds to C1. It does not bind to C2, as shown in the review article. And so this was quite controversial, which made life in the lab quite exciting. And then what Yonggang discovered is that the crystal structure of Chi-B, this is not the structure that binds to the C1 domain. So instead, he found that Chi-B actually has to do something quite rare. It has to switch folds. And you can see that if you just, this is color-coded. So you can see, like, for instance, these two helices become these two beta strands, and this red beta strand becomes this red helix. So you're talking a massive change in folds, and it's this fold that binds to C1. This fold, however, is very unstable. And if you try to crystallize Chi-B, it always crystallizes in this fold, not this fold. And this is why this is being published in 2004, 2005, and this we published in 2015. And it's because of this difference in stability that is why Chi-B binds so slowly to Chi-C. So Gary Chow, former graduate student in a collaborating lab, here he's doing a gel mobility shift assay where you can see that Chi-C, in the presence of Chi-B, slowly forms a Chi-BC complex on the timescale of hours. And it's because, you know, this state of Chi-B, this homotetramer state, which does not bind Chi-C, is so much more stable than the state of Chi-C that does bind. Now, the next question was, well, because Chi-B binds to the C1 ring, so Chi-B binds to the C1 ring of Chi-C, okay? But remember, it's the phosphorylation of the serine residue up here in the C2 ring that allows Chi-B to bind. So somehow there has to be a communication from all the way up here when the serine phosphorylates to all the way down here that allows Chi-B to bind. So that's the question that Yonggang wanted to ask next. And so he again, back to NMR, he looks at the NMR signals coming from the C1 ring, isolated C1 ring. And, you know, so we're just going to follow these two peaks. The dashed lines are just for visual convenience. And so what he did next was he then looked at these two phosphostates of the full Chi-C, one where Chi-C is fully dephosphorylated, one where the threonine is just phosphorylated. Of course, this is just a glutamate substitution. And he noticed that the signals coming from the C1 ring here match pretty much the signals from the isolated C1 ring. And so this state of Chi-C here is the morning state. This is the midday state. And now let's look at the evening and nighttime states where the serine is phosphorylated. Okay, so in both these states, the serine is phosphorylated. And you can see that now these peaks that we've been keeping an eye on have jumped across those dashed lines. And so if you compare all of this, it suggests that when the serine is not phosphorylated, the two rings are not really communicating because the C1 ring signals look pretty much as they do when there's no C2 ring. But when the serine is phosphorylated, it seems like the rings are communicating. And so we said, okay, well, let's call this state where the serine is not phosphorylated the unstacked rings state. And when the serine is phosphorylated, this is the stacked state. And we can actually detect by gel filtration chromatography an interaction between these two rings when the serine is phosphorylated versus when it's not phosphorylated. And so basically, the stacking interaction happens when the C2 ring is tight. Because remember, when the serine is phosphorylated, the C2 ring gets tight. And it's that when it tightens, that these two rings can interact. And so this interaction, the next question is this interaction responsible for Chi-B to bind? So here, Yonggang is looking at signals coming from Chi-B, NMR signals, and here's Chi-B by itself. If he adds just the isolated C2 ring, this is a tight C2 ring, so the serine is phosphorylated or substituted with a glutamate residue. You see that the spectrum of Chi-B looks the same. We already know that Chi-B doesn't bind to C2. If he adds just the C1 ring, he sees a little change, okay? But when he adds the isolated C2 ring that is tight with an isolated C1 ring, you can see now the spectrum changes dramatically, which indicates that Chi-B is indeed binding. And we already know that Chi-B binds to C1. And then when he uses full-length Chi-C with this tight C2 ring, he sees that the spectrum of Chi-B pretty much looks like this one where he added the two rings in trans. So yes, this ring stacking interaction is indeed what allows Chi-B to bind, because this stacking interaction somehow changes the C1 ring that allows Chi-B to bind. And so we want to see if we could mix. Could you try again? We want to see if we could mix Chi-A, Chi-B, and Chi-C together and then watch by NMR the clock oscillate. And so here, you're looking at the functioning clock in an NMR tube, 600 megahertz here at UC Merced. And yeah, we can watch this clock oscillate. And in fact, you can even use two-dimensional NMR and watch Chi-B. This is labeled Chi-B, and you're watching Chi-B over several days, basically binding and unbinding Chi-C. So when you see the signals from Chi-B, it's free Chi-B. When you see the signals disappear, that's when Chi-B is binding to Chi-C. All right. And so we want to look at the rate of ATP consumption by, let me take off my watch here. Okay, so we want to kind of get an idea about the rate of ATP consumption by the Chi-ABC clock using NMR. So here, you're looking at the ATP signal, and this is a mixture of Chi-A, Chi-B, and Chi-C. So the clock is oscillating in the NMR tube. And you can see over time, the ATP signal goes down, the ADP signal goes up. So this is 0 hours, 57 hours, 115 hours. And if you now map out the ATP, amount of ATP, over time, you can see it decreases and the amount of ADP increases. But you can also see that there's an oscillation of the ATP signal and an oscillation of the ADP signal. And so we can actually get at the rate of ATP consumption over time. And you can see that there's this oscillation. And so the rate of ATP consumption is most active in the day, and it's least active at night. And over a 24-hour period using NMR, each Chi-C molecule consumes about 15 ATPs per day. And this matches estimates from non-NMR studies. And we, using crystallography and NMR, we now have this model of the nighttime state of the clock, where in green is Chi-C. This is the C2 ring up here, the C1 ring down here. Chi-B binds to C1. It forms, in fact, Chi-B binds, there's 6 Chi-Bs down here. Chi-B recruits Chi-A to the bottom of the C2 ring. And so this is why Chi-A can no longer stimulate Chi-C, because Chi-A, in order to stimulate Chi-C, has to bind up here. The A loops are up here. And it also, Chi-B binds to this signaling protein KIK-A, and we solved the Chi-B KIK-A heterodimer by NMR. All right, so to finish up my talk, let's just go through just a few slides, asking how does Chi-B switch folds? Because remember, in the daytime, Chi-B forms this state we call now the ground state of Chi-B. It's the inactive state. It does not bind Chi-C, and it forms a homotetramer, but I'm only showing one subunit. At night, Chi-B adopts this fold, which does bind Chi-C, and we want to know how this happens. Now, there's 3 proline residues that are all trans in the ground state of Chi-B, and they become cis in the what we call the fold switch state of Chi-B. This is the active state, the fold switch state. And what we found is that if you ice the sample, okay, basically, and take an NMR spectrum, basically, you can see that all the peaks are in the fold switch state. So this is just an N15 HSQC, and in blue are peaks that represent the fold switch state of Chi-B. And but if you drop the sample into the spectrometer that is set at 35 degrees, so that your NMR sample was initially at 4 degrees, and you drop it in at 35 degrees, what you'll see is that you see these red peaks start to show up. These red peaks represent the ground state of Chi-B, okay? And over time, you can see that the ground state peaks become more strong, and the fold switch peaks become weaker. See that? And so you can actually watch Chi-B switch from one state to the next simply by doing this temperature jump. And what we found is that, so here's a temperature jump. Okay, so here, this is now, these dots here are averaging all the signable residues in the spectrum. And so here, initially, when the sample was at 4 degrees, you can see that most of the signals in the HSQ spectrum belong to the fold switch state of Chi-B. So the fold switch state of Chi-B is more stable in the cold. But once you drop it in to the spectrometer at 35 degrees, you can see that over time, the fold switch peaks get weaker and the ground state peaks get stronger. And if we add in a peptidyl prolyl isomerase, which catalyzes the isomerization of proline residues, you can see the more of this isomerase we add, the faster the fold switching. And so what we found is that the rate determining step in fold switching is prolyl isomerization. And this fold switching is actually temperature compensated. So if you look at the Q10, okay, this is a histogram of Q10 values for residues in Chi-B. And what is Q10? Q10 is the ratio of rates, where in the numerator is the rate at a temperature that's 10 degrees above the rate in the numerator. So the rate in the denominator, the rate in the denominator is carried out at some temperature T, and in the numerator is the rate at the temperature T 10 degrees above the temperature T. And so what you see here is that the Q10 values for switching from the ground state to the fold switch state, remember the fold switch state is a state that binds Chi-C, that it has a Q10 that's much lower than the Q10 from moving from the fold switch state to the ground state. And so remember, clocks have to tell time, so that means that they cannot speed up or slow down with temperature. And so one question of circadian clocks is, how do they not speed up or slow down as the temperature goes up and down? And so here we have evidence that Chi-B participates in temperature compensation in the cyanobacterial circadian clock by having the change from the inactive ground state to the active fold switch state. You can see that this changes much more slowly with temperature, than going from the active Fulcher state to the inactive ground state. And with that, I'd like to acknowledge, of course, David Rice. He's a wonderful colleague, and we have wonderful discussions. We help each other, and he keeps the NMR facility running super smoothly. And I thank him for inviting me to give this talk. And my former group members up here on the top, and my two collaborators, Susan Gold and Carrie Parch, down here. I acknowledge the NIH for their support in this project, and thank you.
Are there questions?
Do we have any questions from the audience on Zoom?
Yep. If not, I have one.
Sure.
Okay, so when you talk about the loose state and the tight state of the C1 and C2 rings, about what does that mean? Are they still bound together?
Yes, when the C2 ring is loose, it's held together by the C1 ring of chi-C. So the C1 ring is tight. And so even though the C2 rings, when the C2 ring is loose and by themselves in the loose state, they don't form hexamers, they don't fly away from each other because they're held there by the tight C1 ring.
Oh, okay.
And remember, each subunit of chi-C is composed of a C1 domain and a C2 domain. And they're linked.
Okay.
Yeah.
Yes. Krish.
Hi, Andy. This is Krish. So one quick question is, when you do the NMR experiment, do you do them in presence of light, or like in Dock cycle, or just all of them are just in room light? What is the experiment?
That's a good question, Krish. So yeah, cyanobacteria use light to reset their clock, but our system, the chi proteins, don't detect light. So once you add ATP, it starts going, and the whole sample synchronizes. So what's really interesting is you have a sample composed of microscopic clock proteins, and they can generate a macroscopic rhythm that you can see with your eye, which is also a very interesting question on how they don't dephase. And as NMR people, you know that you can think of the circadian clock of cyanobacteria. When you mix chi-A, chi-B, and chi-C together, it has a very long T2. It has a very long transverse relaxation time. You can think of it like that, right? It stays phase coherent for weeks. How come it doesn't lose phase coherence is an interesting question.
Yeah. Other questions? Okay, well, thank you, Andy.
Thank you.
Okay, so we'll get on to our next prize, which is a $100 Visa gift card. And so, Dan, take it away. John can.
Hello. Hi.
Hey!
Thank you.
Shelley will make sure that you get the $100 gift card.
Okay.
Congratulations.
Thank you so much.
Okay, so I guess with that, we'll begin our break. So welcome back, everyone. And as we all sit down here from our break, and hopefully everyone's back online. And okay, so let's get started then with the second half of the morning. And our next speaker is Jared Wood, who comes from the University of North Carolina and also from Merck. And his talk will be Chloride, quantitative NMR of chloride by an accurate internal standard approach. You want to get started then?
I'm Jared Wood, so I'm from University of North Carolina, Wilmington. And I'm going to be talking about some of the work that I've been doing, doing a co-op at Merck and also an internship that I did this past summer. So this is going to be on not only chloride NMR, but also fluorine. So I'm going to get into the chloride part first. So first, to talk about chloride QNMR, I'm going to introduce the concept of counterions. So counterions are commonly used in active pharmaceutical ingredients to improve upon their solubility as well as their bioavailability and stability. I have a really common example here just to show you. This compound at the top is pseudoephedrine. I'm going to get a pointer here. Here we go. So this is pseudoephedrine up at the top. It's not quite soluble in water, not nearly as much as the hydrochloride salt version of this drug, which is Sudafed, and that dissolves much better in water. So chloride specifically is a very popular counterion. It accounts for being used in approximately 15% of approved small molecule drug substances and many, many more that are not approved. And there are many different methods that are used for QNMR, specifically for analysis of chloride, not QNMR. And these are some of those methods that are currently used that are totally inferior, and you know that because we're at an NMR conference. So a few are including HPLC and other forms of chromatography, and it is widely known that with chromatography comes method development for each and every individual compound that you're working on, and also you need much more reference standards specifically to use the same one that you're trying to quantify. And also there are some other methods like titration and ICP, which are known to have some specificity issues. In particular, ICP is known to be nice for quantitation of chloride, but is not able to distinguish between chloride on different molecules. And that's something that QNMR is totally capable of, and we're going to talk about that here in a bit. But again, I'm preaching to the choir here. I've been doing NMR since late 2020, and I can see why you all like it so much. QNMR is great because with our methodology, you know, it's robust, sensitive, accurate, and precise, and it's used for all sorts of things like assaying concentrations of heteronuclei, assessing purity, and monitoring reactions. Specifically with NMR, it's really nice because it is often used as a qualitative technique, of course, and you can find impurities, find out exactly what they are, and with QNMR you can find out exactly how much of those impurities are that you have performed structural elucidation on. Pretty simple method. You just need to know the purities and molecular weights of your compounds and do some simple integration to find out how much you have. In QNMR, chlorine is a particularly special nucleus to work with in comparison to something like proton or carbon, which have a spin of 1/2 and much better frequency ratios or gyromagnetic. They have quadrupolar moments of 0, where chloride has particularly significant quadrupolar moments, which is referencing the asymmetry of the nucleus. It's lower on the periodic table, and this nucleus asymmetry causes an issue with fast relaxation, which then broadens out your lines to the point where you can't really integrate them and perform an accurate quantitation. However, this is something that's not really reported on that much and is very interesting. But when you have an ion or molecule that is symmetrical, like these three shown at the top, chloride, carbon tetrachloride, and perchlorate, they are fully symmetrical in 360 degrees, and this alleviates the asymmetric stress on the nucleus, which sharpens up those peaks and makes them totally viable for QNMR analysis. These three at the bottom, they are symmetrical in some degrees, but not fully in 360 degrees, and they look terrible. Cannot do it, cannot work with it. there's been some prior work done on chloride QNMR specifically. There was an external standard method that was reported in 2006, which it reported some good linearity data, but not any accuracy or precision data. And then there's been application of Poulhazan as well. There's actually a fairly recent paper in 2022. They didn't report any precision, but they still had some relatively nice accuracy. It was around the 3% error amount. And this is totally fine if you want to do something like assign a mono versus a bis salt. It's totally accurate enough to do that, but it's not particularly great for quality control purposes and things of the like. Our method, far more accurate, and we wanted to assay these hydrochloride salts here so that we could validate that. These are all nice salts to work with, little to no hydroscopicity, except for this one compound here, polyethylenamine hydrochloride, which is pretty hydroscopic, but we wanted to use that as a test molecule because it is pretty popular and is used for transfection, and we thought it'd be a nice thing to try quantitation on. So our internal standard method is pretty simple. You just need your hydrochloride salt. This is particularly for quantitation of hydrochlorides, and also pretty much anything that has a individual chloride ion. And our internal standard choice is potassium perchlorate. We dissolve that in 1:1 deuterated acetonitrile and D2O, and we're able to do an accurate quantitation. Now, why did we choose potassium perchlorate? It meets a lot of our criteria for what a really nice internal standard is. There's a lot of reported criteria that people talk about, like 1, it doesn't overlap with the resonance of your analyte. 2, non-hydroscopic. 3, soluble in whatever solvent system you're working with, has similar relaxation properties to your analyte, and so on and so forth. It's very nice, particularly because potassium perchlorate, unlike other perchlorates, is non-hydroscopic. So only work with this one, no other ones. Sodium is very hydroscopic in this case. So we put together this solubility matrix so that we could see what the best solvent mixture is for dissolving your hydrochloride salt and your internal standard. That's with potassium perchlorate, and we wanted to verify not only the solubility in these solvents for just the drug substance alone, but also with the internal standard. Okay, so something that is also not reported on too much in the field of QNMR is verification of the co-dissolution of both your analyte and standard in whatever solvent system you're working with, because it is very important to do that. As you can see here, I have these boxes indicated with these colors to show just how soluble these things are. And when adding the standard to the mix, the solubility overall does decrease. So it's important to note what does and does not dissolve with your internal standard. We found that the best mix overall is a 1:1 ratio of D2O and deuterated acetonitrile. It is the same in both cases here. We have black boxes just for undissolved to 17 milligrams per mil, gold boxes for higher than that, but not as high as twice as much, and then cyan boxes for the best overall. Next, solubility effects. We wanted to see how these solvents looked just for peak shape purposes and signal-to-noise ratios. So with perchlorate, the best results overall did come from the 1:1 mixture of deuterated acetonitrile and D2O. And for chloride, it seemed to get only better as we moved D2O up to 100%. But as you saw on the last slide, the solubility in 100% is not as good. So 50-50 is still the best here, and this peak shape seen here with chloride at 50%, that is still totally sufficient for quantitation and is still giving us very good results. We also looked at temperature effects. Specifically, temperature at 65 degrees Celsius over 3 days was tested, and that did end up showing some pretty stable results, but we do want to point out that it is still easier to use room temperature with a given method, and you also want to do that to avoid any potential degradation of whatever pharmaceutical substances you're working with or whatever it is you're working with. And we still do get the best peak shape with 25 degrees Celsius for perchlorate, and it's still also totally sufficient for chloride. So that's what we went with, room temperature. So optimization of the acquisition rate. Looking at the T1s here for both chloride and perchlorate, you can see they're really small in comparison to what's typical for something like a proton T1. And with that, we're actually able to bring the acquisition time and relaxation delay down a lot. We wanted to start out with 2 seconds and 1 second respectively for those two, and look at the area to area ratio for the chloride and perchlorate peaks. We were starting out at an experimental time of 103 minutes, and we were just doing some initial optimizations. We were able to bring that down to about 21.5 minutes for an experiment. Very nice because, again, chloride is not a particularly sensitive nucleus. So these are our overall method parameters, generalized for users of different instruments and software. I've gone through these already. We used a standard 90-degree pulse sequence for 35 Cl, that isotope that is more abundant, has a better frequency ratio. And we have this relaxation delay of 0.1 seconds, acquisition time of 0.5 seconds. The transmitter offset needs to go directly in between the two peaks, right at the halfway point. This is a very large spectral range that we're working with here, as you can see at the bottom. So chloride is showing up at 0 ppm, and perchlorate is all the way at 1,000. You may have concerns about the 90-degree pulse over that really large bandwidth, but I'll get into that later. So our spectral width, 1,200, number of scans, 2048. And for data processing, we used a 20 hertz line broadening and a Whittaker baseline correction. So for those of you familiar with baseline corrections, specifically Whittaker is infamous for being terrible. It's overly aggressive. And in our case, it's actually really nice. We don't recommend it for anything else, unless you have completely verified that it works the best, and that it does here. This is how the spectrum looks, and it is very accurate, and here we have the results to prove that. So here I'm showing absolute error for all of the different compounds that we worked with. We did 3 runs of each and an average showing precision data and accuracy data, and our accuracies never got any worse than 0.55% absolute error. And I've also included these external standard measurements from this 2022 paper that was applying Polcon. Those are roughly around 2.5% absolute error. And I'm not cherry picking here to make our data look better. This is totally representative of what an external standard method will get you. We've also tested this across multiple instruments and at different concentrations just for robustness purposes, and it is still very nice. So further on robustness of the method, we had initially optimized the pulse width to 21.5 microseconds, giving us this 0.5 area to area ratio. And also, we wanted to see how shifting the transmitter offset from the midpoint of the 2 peaks affected the area to area ratio. And just moving it by 0.5% gives us 1% added error into our measurement. And going all the way down to 10%, that gives us 27% error. So definitely keep it in the center. And if you haven't heard of this applet up here at the top right, it's pretty neat. You can write that down or take a picture of it if you want, but if you enter in a pulse width and spectral range, it is going to give you an excitation profile for you that looks just like this. And it's very useful. It's showing us that if we put it in the center and we go to, say, where the perchlorate peak is or where the chloride peak is, we're getting about a 45% of that 90-degree. And if we were to bring that pulse width down to 15 microseconds, that brings it all the way up to 70%, which is much, much better. Looking at these results here, it significantly improves upon the signal to noise that we were getting with the method, and also decreases the effect of shifting the transmitter offset. Still, you certainly do want to put it in the middle, but in the end, this is the best for the method that we had worked on. So 15 microseconds pulse width. And lastly, we did also try to utilize the chorus pulse sequence, but in the end, it did not end up being useful because both chloride and perchlorate do have a large T2 relaxation difference, and that did hurt our signal-to-noise ratios. But still a notably nice method, just not for this. So that's it for the chloride work, and next I'm going to get into some further work that I've been doing at my current Merc co-op position. We're trying to do fluorine QNMR. Much nicer than chloride. It is much more sensitive, has 100% abundance, 1/2 spin, no significant quadrupolar moment. It's 0. Very nice. We wanted to, of course, pick an internal standard that meets all of the criteria you want, again, for different types of analyses. We found this compound, 5-fluorouracil. Is commonly used as an oncology medication, and it has a really nice fluorine chemical shift, great for quantitation. It has a proton chemical shift as well, which is in a spot that does experience a lot of resonance overlap. It's about, I think, 7.2, something around there. But it is still, it has a very nice shape and is great for integration if it is isolated. The compound itself is unreactive, incredibly soluble in DMSO, sparingly soluble in water. And this is an interesting point because when we were working things out to find this compound, we wanted to take a look at a list of internal standards that already exist. So Sigma Aldrich has a list of certified reference standards for QNMR called the Trace Certs, and they don't have anything for fluorine, interestingly enough, that dissolves in water very well. So we thought it might be a good idea to find something that does dissolve in water and tell them to start developing it. It's a nice T1 as well. We did some initial work with this compound already for quantitation of fluconazole. And with the proton peak here, we were actually able to nicely get an accuracy value of 99.8% when performing quantitation. Great results already. With fluorine, not as nice. Putting a transmitter offset right here in the center between the fluconazole peaks and fluorine peak for 5-fluorouracil, our accuracy results just for that with a 90-degree pulse were 101.7% accuracy. Certainly could be improved upon. And we did improve upon it later. Whittaker Smoother actually works nicely as a baseline correction here as well, because we have this rolling baseline that we get when acquiring the spectrum. Of course, this could also be solved by removing the first few points of the FID and doing some prediction to fix that, but we didn't do it. Haven't tried it yet. lastly, we also tried a short flip angle when putting the transmitter offset on the 5-fluorouracil peak. And that gave us a really nice accuracy of 99.2%. So that is within plus or minus 1% and is very nice for fluorine quantitation, but we can certainly do better. And that's it for 5-fluorouracil. So now that we've been inspired by not seeing a fluorine standard that dissolves in water for QNMR, I wanted to go through as many possible structures as I can to see if there's anything out there that does exist that will work really nicely. So what I did is I searched through thousands of structures in PubChem, and I'm still doing it now. And what I would do is take these structures, if they looked interesting, I would do some instant predictions of data in ACD labs software, specifically for proton chemical shift, fluorine chemical shift, and also water solubility, which is something that it can predict. It is useful, and we have a few structures already, and I wanted to present some of the data on that. So here is a solubility chart again for some of the compounds that we have tried out, and this is in a pretty wide polar range of solvents. As you can see, we have chloroform spanning all the way to D2O, and a couple of these compounds do have nice solubility in all of them. That would be trifluoroethanol and also hexafluoroisopropanol. So we're going to do more to try to find out a better standard because those two are actually liquids. Hopefully we can find a solid that dissolves in all of these solvents, because now I'm starting to think it's possible that we can find something that works for everything. I'm hopeful. And we wanted to also present these ACD predictions and also actual experimental values for chemical shifts. And I have these green areas here, specifically from 9 to 10 ppm and also from 5.5 to 6.5 ppm. Apologies to any folks who are colorblind. I wanted to point out those regions. I consider these nice regions that do not experience a lot of spectral overlap between multiple resonances. And these are optimal places for proton chemical shifts to be. And the nicest chemical shifts come from 5-trifluoromethylpyrimidine and this 3,5-dinitrobenzotrifluoride compound. So I'm going to keep optimizing based on these criteria, and I'm going to keep searching for unique structures that could potentially be used for a dual quantitation through both proton and fluorine QNMR analysis. It'd be really cool to have something that is, well, can be orthogonally used to do that, that dissolves in a wide array of solvents, right? So that's pretty much it. So in conclusion, we have a robust, accurate, and precise QNMR method to assay levels of chloride. And I have this linearity chart here at the bottom just to show some linearity data. And as for the fluorine QNMR stuff, we're still working on it, and we're incredibly hopeful that we're going to find something amazing. I am especially, and that's it. If you have any questions, I'll be happy to answer them.
So, any questions? Yes.
Yes.
Awesome presentation.
Yep.
Good
Yeah.
point. Awesome presentation.
You got to hold it close.
Awesome presentation. Thank you. I'm curious about the potential, ignoring the simplicity of using a 90-degree pulse.
Mhm.
I'm wondering about the utility of putting a shaped pulse in. You mentioned that you tried chorus and weren't seeing a big improvement. I'm wondering for both the chloride and the fluorine, I'm wondering if you'd get actually better results with the shaped pulse.
It's totally possible for fluorine, I think. But chlorine specifically is interesting because, of course, the differences in T2 relaxation between all sorts of things, because this was very specifically developed for analysis of anything that just has a free chloride ion. So anything like that should have a particularly different value from perchlorate, which is our internal standard. But that's what it was developed for. But it's a very nice point to bring up, and we'll probably look into it.
Krish?
Simple but excellent talk. Two questions I have is, for your chloride detection, the charge state of the chlorine is important. Did you try as a function of pH, the function of with D2O, how the peak changes?
That is a good question. We didn't do that at all.
Because, you know, in water, it does form the ions, so that could be an interesting factor to look at.
Right.
And the second on the fluorine data is trichlorofluoromethane is a known standard for fluorine at 0 ppm. Did you try how does that compare with your current internal standard?
We did not try that one, actually. We didn't actually consider anything that was coming from any trace cert lists or anything that anyone has already reported as a proton standard that has fluorine on it either. We did try just like TFE and hexafluoroisopropanol exclusively because they were reported. But I also will add the point that those, while they do have protons on them, those are protons in awful regions of the proton spectrum and should not be used for quantitation. But no, I did not try that compound. But it's a good point to bring up, yeah.
Hello, I love the chloride work. I'm questioning about the application of it to the API. So you can accurately determine the amount of chloride, but is there any concern for how that relates to the API itself? Perhaps if you have multiple salts that co-precipitate, or if you have a contaminant like water or something else that test will be blind to. I'm wondering if you could comment on that.
So, are you possibly getting at? It's a good question. Are you getting at something that could be answered by the fact that only symmetrical molecules will work? I'm not sure I fully understand.
Let me rephrase. So, if I understood correctly, the chloride ion is being used as a stand-in for quantifying the cation component of it.
Yes.
So what about situations where you have multiple cations that will precipitate as a hydrochloride salt? The chloride test will tell you how much chloride is there, but how do you assess the accuracy of that for the API? Whether you have a contaminant that came in that's blind to chloride as a nuclei, or if you have some side reaction that's also producing a salt that might still have a chloride and contribute to your test. But if your result is being used to say about the quantity of the API itself, how do you assess that your accuracy carries through from chloride all the way through to the organic molecule?
Yeah, so, yeah, that is an excellent concern. The hope is that generally you wouldn't be using mixtures of things to begin with, but it would be readily apparent when calculating just how much chloride you have, and then therefore how much cation you have when your molecular weight is not representative at all of the values that you're calculating just based on integration. So it would be best to definitely separate everything you can individually when doing this QNMR analysis, because, yeah, NMR is not going to distinguish between chlorides belonging to different hydrochloride salts.
We have Daniela Robilar, if she'd like to go ahead and unmute herself and ask her question.
Hi, thank you for your talk. I have a quick question for the chlorine NMR. The hypochlorite was terribly bad. You just mentioned that as it wasn't, well, it's linear, it's a linear compound, so the peak would be not that sharp, but it's really, really bad for discarding it as an internal standard, internal calibrant, sorry.
I'm sorry, could you repeat the question?
Yeah, my question is if the hypochlorite, did you test the hypochlorite as internal calibrant? Because you just mentioned that the symmetry wasn't good, so that would give a bad peak. But I just want to know if you try it and the results that came from that try.
So hydrochloride it does have nice chloride line shape in QNMR. Hypochloride. I'm sorry, I didn't hear that. The echoes in the room. No, we did not actually try that, but I know that the people I've worked with historically have seen that in the past. I have not experienced that for myself.
Okay, thank you.
But generally, things like that do have, because of their asymmetry, pretty bad line shape.
Okay, thank you.
Nice talk. Couple of things that I wanted to ask about.
Sure.
In your observation with the offset, where the position of the transmitter frequency is, and then as you moved, your error was higher. That's expected, and it can be calculated because you actually showed a picture of the applet result of. It's primarily for a rectangular. It's just based only upon the pulse width, right? We can back calculate. So having the offset exactly at the center doesn't give you anything more than having it 1,000 hertz and calculate what happens when you push it by 1,000 hertz. It's a block equation. So have you tried that to make sure that that error you are seeing is really the offset excitation profile error rather than something else beyond that? That's question number one. So because that may not be an issue, having the offset exactly at the center may not be an issue because it is an equation, nothing more. The second point that I just wanted to bring up is more like a comment which came up with the last question as well. You know, when we talk about asymmetric chlorides having a line shape, and we are really talking about broad lines. Line width are decay rates and so on and so forth. Line widths, in my opinion, maybe I'm a minority here, should have nothing to do with quantification. So with that, I'll close.
Right. Yeah, those are both good questions. So for the second one, the primary concern for quantitation is being able to, so let's say hypothetically that it's so broad that you don't even know where the baseline is relative to where the actual top of the peak is, because if it is broad enough, it might actually be difficult to find where it actually is touching the baseline. So you might not know exactly where to integrate it. Perhaps you could argue that, oh, maybe if I just integrate over a wide enough area anyway, then I should capture all of what's underneath that peak. But this is mostly for the concern of being able to find out exactly where it hits the baseline.
So it's really a methodology that you are using integral as a surrogate to quantify.
Yes.
So it's not about the quantification aspect itself. It's the methodology that you're using to quantify dictates that you can't use wide lines. That's it.
And it's also crucial to our signal to noise to have something that we can easily see, because that does have a significant effect on the integration that we get. Now, I'm really sorry, I think I might have forgotten what the first question was about.
The first question was about the offset. Having the offset off simply means you just need to add one more equation to your quantitation. That's pretty much it. So, particularly if you are using simple rectangular pulse, which is easy to calculate, the offset effect, offset excitation profile. You had an applet. It's known for years, right? So.
Yeah, right. So we did experimentally verify the effect of moving the offset around, but we did not perform any calculations specifically to see how that did affect it.
Thank you.
Let's get on to our next prize. And with the third prize, we have a wonderful Ivan cutting board, which everyone wants to have. I have one. I just couldn't bear to actually use it for a cutting board. See, but you can use it the underside, but I prefer to display mine. But let's get started with the, see who gets the cutting board. Luc Tremblay. In this room? Oh, bad. Okay, well, let's try again. Oh, okay. Anna Drumick.
Yes, I'm here.
Hooray!
Hello.
Congratulations.
Thank you very much.
Shelley, she will get your address and send it off.
Thank you very much.
Okay, good. Okay, well, let's get on to our next talk. Our next speaker is introduce Marc Taraban, who is online. And he's going to speak about water proton NMR for analysis of pharmaceutical products.
Thanks a lot for inviting me for present at Ivan Conference. It's a great honor for me. And I'm here representing University of Maryland and also the Institute for Bioscience and Biotechnology Research, which is the joint venture of the National Institute of Standards and Technologies and University of Maryland. The building you can see behind my back as the background. So let me share my screen, and I hope you can see it. Okay, and so my today's presentation will be just a short review of the work our group has done on the application of the time domain NMR for analysis of the biopharmaceutical products in their original containers without taking them out or sampling out. We call this technology water proton NMR. This term has been coined by Bruce Yu back in 2017 in our analytical chemistry paper, and we call it WNMR. And this technology that we are developing have some similarity with MRI, The signal source is also water molecule, which is the major solvent for biopharmaceutical materials, biopharmaceuticals. And water NMR allows to collect data on a drug product non-invasively or directly in a process flow. And it's a contact-free and gives you the possibility to do the process flow monitoring, and it can allow you to inspect drug product which remains intact for further use. After the inspection, you can give it back to patient. And the water NMR uses compact, inexpensive, wide-bore benchtop NMR instrument, demonstrated the sensitivity to API concentration, aggregation, particle clustering effects, and as I mentioned, could be used in a static and a dynamic flow mode, which is useful for the continuous biomanufacturing too. Here you can see the benchtop NMR family that our group is using. We have a benchtop instruments which allows you to do the measurements under controlled temperature with the variable temperature from -80 to +80 centigrades. And we also have a flow NMR system which allows you to mimic biomanufacturing, continuous biomanufacturing settings. We have a standard NMR spectrometers with the no temperature control, where we try to keep the magnet temperature close to the room temperature. So these instruments are standard at 18 centigrades controlled cold room to have a temperature of the environment at least 5 to 7 degrees lower than the temperature of the magnet for the stabilization purposes. In all instruments, we have a capability to put the whole vial or the whole pen or whole drug product container inside the instrument to do the measurements without opening, because of the wide bore of those instruments varies from about 18 millimeters to 50 millimeters. And what exactly is measured by water NMR is just a standard time domain CPMG pulse sequence, and most often used at a fixed tau interpulse delay. Variable phases of the pulses sometimes are employed by different software of the different instruments. And we also have a capability to do the automatic variation of the tau interpulse delay for some multivariate experiments that I will be talking later. In general, the result is just a simple single exponential echo, spin echo decay, which is used to extract relaxation rate of water. And this is our identification parameter, which is used for our analysis. Why the reasons, just let me briefly mention the reasons of water sensitivity to concentration, to different clustering effects and different mechanisms that are controlling the sensitivity of water. One might just select 3 different mechanisms. For example, in case of the protein aggregation, here it results in the large assemblies with faster relaxing protons, and water molecules involved in the proton exchange with these protein molecules will also have faster relaxation rate compared to the bulk water molecules which are not involved in this exchange. Another mechanism is magnetic susceptibility contrast, which allows to creation of the magnetic field gradient and the solute and water molecule interface, where the water molecules will relax faster. And the third mechanism generally seen in the clustering of nanoparticles is the formation of the cavities inside the clusters, with the diffusion exchange of water molecules coming of those clusters also having much faster relaxation rate compared to the other water molecules not being in these cavities. All of these 3 mechanisms contribute to the different point of applications of water NMR, a different lifetime of any drug product, starting from formulation, where you can study drug stability, drug product stability, and to the manufacturing step for the inline measurements of the concentration and formation of aggregates, and up to the quality control at the very end life of the product, even at the point of care during the release of the product or at the very point of care, for the injection to the patient. So here, for example, is an example of the proton exchange sensitivity used to explore the stability of the drug product, therapeutic antibody, monoclonal antibody formulation. Here, the response, we can see the linear response of the relaxation rate to the different levels of soluble aggregates formed at different stresses during the freeze-thaw stress, heating stress, and agitation stress of the therapeutic antibody formulation. And relaxation rate on the right panel is also shown the sensitivity towards the formation of the aggregates of this therapeutic antibody over the wide range of stress levels, and responded differently to different stresses. You can see here that for heating stress, we have a much stronger slope compared to the freeze-thaw stress, and for agitation, we have a non-linear behavior because depending on the stress level, the aggregates might dissociate. So we also have demonstrated the capability of water NMR for sensitivity towards the concentration and the aggregation in the process flow. We have developed the benchtop flow NMR instrument where we have mimicked the continuous biomanufacturing conditions, and we observe the sensitivity of our instrument to the flow rate depending on the flow cell configuration. For the linear flow cell and for the coiled flow cell, you have a different slopes of the dependence of water relaxation in a pure water sample depending on flow rates. This will allow the user, for example, to detect the abrupt flow rate changes or jumps during the in the conditions of continuous biomanufacturing, to see the changes in the flow rates and monitor them using the flow NMR. We also have demonstrated the sensitivity of the flow NMR to the concentration changes for two different proteins at different flow rates. You can see here in these three-dimensional plots that you have essentially planar or linear flat plane that allows you to monitor the concentration changes under the continuous biomanufacturing settings. We also have shown that the flow NMR is not only dependent on the concentration, but it also shows the dependence of relaxation rate of water on the percent of the soluble aggregates detected from the SEC and put inside the instrument just for detection of the dependence of R2 versus the flow rate and aggregation concentrations. All of these measurements are done non-invasively, and all of these measurements are inline, real-time, and contact-free. That allows this instrumentation, uses this approach under the biomanufacturing conditions to monitor flow rate changes, monitor concentration changes, and monitor the aggregation of the API in the process flow. For the quality control at the point of care, for example, this technology might be used also to understand the uniformity of the different drug products. Here in this experiment, we have done the analysis of the 4 different insulin products for the patient's self-injection, presented as a pre-filled insulin pens. And here we have analyzed 2 pairs of the innovator and the follow-on product like Lantus and Basaglar and Humalog and Admilog are the corresponding pairs of innovator and follow-on products respectively. And we analyzed this, all analysis can be done at 4 centigrades in the temperature-controlled NMR, and of course without taking anything out of the pen, right inside the pen. And these results show that for all 4 insulin products and for all 10 pens of each product measured, we have pretty much good uniformity with the very small variation of different pens from each other. And note that there is a difference between the follow-on and the innovator products in these two pairs. The relaxation rates are different. So our relaxation rate of water is also capable to distinguish between innovator and follow-on products, which supposedly should have identical chemical composition. Also important implication of the, for example, one of the mechanisms of magnetic susceptibility contrast, we have used this to understand the potential of using for the drug product stability for aluminum adjuvanted vaccine. Aluminum adjuvanted particles have significantly larger magnetic susceptibility contrast in addition to the OH groups on the surface. So the sensitivity of the aluminum adjuvant suspensions to changes in to different changes in stability and sedimentation protocols demonstrate extreme sensitivity of water relaxation rate. So two important parameters of drug product stability here have been explored is the sedimentation rate, which tells us how strong is usually the adsorption of the antigen on the surface of the adjuvant. And also sedimentation volume ratio is the ratio between the sediment and the whole suspension that tells you about how compacted is the sediment in aluminum adjuvanted vaccine, and tells about the possibility of pure and full resuspension of this drug before the injection. So all of these measurements are done non-invasively and in the original closed container without taking it out, and it gives the idea, the possibility of measurement of the sedimentation rate, monitoring the changes of relaxation rate on time, and understanding of the difference between the contributions of two parts of this product, fully supernatant and the sediment, to the relaxation, to the decay of the echo signal, which is in that case will be bi-exponential and will allow you to get the contributions of two different parts of your sample. Another very important problem of the modern pharmaceutical biomanufacturing is the understanding of the ratio between a full and empty gene therapy products. In this exercise here, in these experiments, what we did, we analyzed the possibility of water relaxation rate to distinguish between empty and full capsids, with capsids full with DNA or gene material. And from this experiment, you can see that in case you have an identical titer, identical viral particles concentration, you can see the significant difference in relaxation rate of the empty capsids and the full capsids. And this allows you to get there, if you do the titration and see and mix two samples of the full and empty capsids in different ratios, you can see there is a significantly, it's not significant, it's a absolutely ideal linear dependence of the relaxation rate on the percent of the full or empty capsids, and it demonstrates the capability to determine this non-invasively without opening the vial, which is extremely important because right now the measurements of the full versus empty capsids is a pretty long procedure, requires a lot of efforts, and of course all of these measurements are invasive and they are you are losing a very expensive samples. In these experiments, you are not even opening this vial with the capsids, and you can give it back to the manufacturer or even to the patient. These drugs are extremely expensive, and one of the market is about $2.5 million per injection. So we also used our technology to fingerprinting of the drug product, specifically to fingerprinting of different vaccines. And all this analysis has been also done in the drug product vials and drug product containers without sampling them out. Here we have analyzed different vaccines that contains aluminum adjuvants in the same concentration, similar concentration, which might be some kind of the very close to each other. These two vaccines, for example, have almost very close values. These two vaccines have identical values of aluminum adjuvant. And we also analyzed two vaccines which are non-adjuvanted. The idea was to probe whether the water NMR is capable to do the fingerprinting because of other components of those vaccines and to understand the sensitivity of water NMR to the unique content of the vaccines. We used to do several different approaches for that. One of them we called univariate, where you essentially analyze relaxation rate of water at a fixed parameters of CPMG, like at a single interpulse delay tau value. And you also might have a dependence of relaxation rate on the interpulse delay, and you can look at the relaxation rate in time to monitor the kinetics specifically necessary for the sedimentation kinetics of the different vaccines. And you also might use both approaches at the same time. For example, you can monitor changes in the vaccine over time, and at the same time vary the tau value. And in that case, you might have a unique fingerprint of this vaccine. Here, for example, in this first example, we used the univariate approach and multivariate approach to analyze two vaccines that are non-adjuvanted, and we compared them with physiological saline. And you can see here that if you do the measurement of the single tau value at 500 microseconds, you have a pretty close relaxation rates of these two vaccines. They are almost non-distinguishable from each other, and they are pretty close to the saline. But if you do the same measurements with the varied interpulse delay tau, you can see significant difference between these two vaccines, and also the difference between these vaccines and the saline. And that allows you to get a fingerprint of these two vaccines for their analysis and to find out the counterfeits, for example, if these counterfeits are just a simple physiological saline. Here, this is application of the univariate and multivariate to the adjuvanted vaccines presented in the syringes. For example, here, in the fully dispersed vaccines, so the suspensions is fully dispersed, and here you can see there's a certain significant difference between these two vaccines, and they're significantly different from physiological saline. And even in the sedimenting state, the values at the very low values here, the very low values of tau, this sedimented vaccines demonstrate very similar values, but as soon as you will do tau dispersions of them, you can see the difference between these two vaccines. Here, two vaccines are produced by the same Daptocel and Tanivac, are produced by the same manufacturer, and they contain the same identical concentration of aluminum adjuvant. And you can see how close they are, both in the sedimenting state and the dispersed state at a single tau value. And when you do tau dispersion, you also can see very close similarity of these two systems, two vaccines. However, if you monitor the sedimentation kinetics of these two vaccines, you will see that they are significantly different from each other. The rate of sedimentation of Tanivac is much larger compared to rate of the sedimentation of the Daptocel vaccine, and that allows you to distinguish these vaccines from each other. But when you do the application, of this multivariate approach, together with the variation of the tau value, you can see that these two vaccines are even more different from each other. Here on this three-dimensional plot, you can see that Tanivac and Daptocel demonstrate totally different dependencies of the sedimentation kinetics together with the variation of the tau value here. We also, in our experiments, we can do, in our protocols, we can do the variation of the tau in cycling way. For example, during the sedimentation of the Daptocel, for example, we applied this procedure in a way that we are changing tau in ascending way and then in descending order. So essentially, you can have this kind of cycling note that when the vaccine sediments are relaxation rate is dropping, but the increase of the tau results in the growth of relaxation rate. So it's essentially somehow slows down this decay compared to the sedimentation under the conditions of the single tau value. But this thing is totally unique fingerprint of the changes of relaxation rate, and it can be used for the fingerprinting of these vaccines and use them as the for the detection of the potential counterfeits. And it's even more visual if you look at this in three-dimensional space. It's even more visually differentiating the single vaccines from others. So with that, I will summarize my talk and mentioning the possibility of the use of water NMR for quality and stability monitoring in pharmaceutical products in their original containers, whether it's be sealed vials, prefilled syringes, or injection pens. And flow water NMR would be used for the contact-free real-time inline process analytical technology for continuous biomanufacturing. Water NMR could be uniquely used for analyzed gene therapy products and allows to quickly and reliably estimate the ratio between viral capsids filled with the gene therapy products and empty ones. And also, in the future, we envision the possibility of the very small compact NMR instruments that will be also could be used in the point of care in the pharmacy or the healthcare facility, and even by the individual patients to ensure the quality of the drug prior to its administration. So with that, I need to acknowledge the help and great collaboration with my friends and colleagues, Dr. Briggs, Professor Yu, our students, Yiling Wan and Pratima Karki, Mike and Jones from Pfizer, and Dr. Grunin from Resonance Systems, great help from our colleagues from the companies, Oxford Instruments, Resonance Systems, and MRR. And also, I should acknowledge the help from the funding from NIH, NIMBL, Maryland CERSI, Pfizer, and the University of Maryland Strategic Partnership. And since I cannot be here today with you, here's my business card that you can see my information, my contact information, and feel free to contact me anytime when you will be interested to collaborate. And with that, I thank you for your attention, and I'm open for the questions.
Thanks very much, Marc. Okay, so our next prize is, it's not the, it's a $50 Visa gift card. And so let's take it away, Dan. Leopold Merloff. Uh-oh, okay, he loses. Let's make sure that's... Two, 1, 0. Nope. Now he loses. Claire Dickinson. Uh-oh. Friends don't count here. You have to be present. 3, 1, 0. Bing ran. What? Oh. Oh. Friends or husbands. Wow, this is getting kind of. Well, 0. He lost. You can get to tell him. Yeah, but actually, though, yeah, we could, if you had made it in time with a text message, maybe, but. Okay, now we're going again. Oh, Kun. Yay! Okay, so with our prize given, we're up for our next event, which is the long-awaited Ivan Founders Award. And to do that, John will come up and give the award and reveal the winner.
Monterey Peninsula, Pacific Grove, Asilomar. What's not to love, huh? It's too cold here, but I was down in Los Angeles a couple of days ago. It was 55 degrees down there, so we're not alone here. But anyways, my apologies for being tardy this morning, but better late than never, as they say, and welcome everyone. Really appreciate everyone turning out today, both in person and online. Looks like that our technology is working out pretty well today. No glitches or hiccups, from what I understand. I know that there were some introductions a little bit earlier this morning, but I just want to go around the room very briefly. Want to give thanks to my fellow Ivan Board of Directors members, Dave, Dan, Krish. There's Dan right there, and of course Eric and Jake back in the far corner, our production crew. Shelley Hammond, who has basically put together all the logistics of this, not only meeting today but all week, and also Sharon. I don't know where. Maybe she's outside somewhere. But anyways, once again, everyone welcome, and I think we have a very exciting part of the day today. And that would be, of course, the 2023 Ivan Founders Award. And the award is now in its 3rd year and is given annually at the Ivan NMR Users Group pre-ENC meeting. The purpose of the Ivan Founders Award is to recognize an individual who has demonstrated excellence in support of the magnetic resonance user community with work and service above and beyond the normal scientific contributions and consistent with the mission of Ivan. The award committee received many worthy nominations this year, reflecting the goals and mission of Ivan, but one individual stood out among the rest with their significant service to the global NMR community. Ready? Drum roll. Ron Crouch, if you would, front and center. Ron, the Ivan Board of Directors and the Ivan Founders Award Committee would like to recognize you for the significant impact you've had in your career on the global NMR community through your wisdom about NMR methods, your willingness to engage in personal help, collaborations and training in the practice of NMR that go beyond the usual scientific expectations of your employers and customers. A statement from your nomination is, and I quote, "Over his long career, Ron has repeatedly demonstrated that his primary strength and contribution to society is not just his innate ability to recognize a methodology and its potential impact, but then to follow up with passion and to bring those tools to novice users through implementation and education." Ron, we're very proud to present you today with the 2023 Ivan Founders Award for your community service. And we have this, well, it's two pieces, so you got to be careful. And I'm just going to slide it over there. It's engraved with the Ivan logo, Ron Crouch, and Ivan 2023 Founders Award. And maybe even better for a rainy day is a check for you for $2,500. Ron, congratulations. Job well done. Very happy to have you with us here today, and we hope you'll accept the award.
It's a humbling honor to accept this award, really because of who it comes from. This community is born of a love of NMR and a love of everything about the technology and the people that are in the NMR family and the support of it. And for me, it's a fabulous, fabulous, humbling experience. Thank you so much, John. I appreciate it. My roots in NMR go back very far, all the way back to the 1970s in the pharmaceutical industry. And while there, as a part of that family, I met many wonderful people along the way. Remaining today as a very dear friend from my NMR Foundation days is Gary Martin, and we still talk NMR virtually every day. He has a real love and excitement for the technology, and it's just something that is just contagious in the NMR community. And I thank every one of you for what you've done. Then we moved on to the Varian NMR family, and I see some Varian orphans that are here with me today from the past. This award is as much theirs as my own, because I learned so much about NMR technology from interacting with Krish, Dimitri, Dan, Lima. I even learned what solids was. So I must thank everyone. I humbly accept the award. Thank you.
Congratulations, Ron. How was lunch for everyone? Good. You're entirely welcome. Looks like all present and accounted for. Next, sort of a mini session, but very, very important mini session. We all know and love our favorite helium crisis, 4.0 as it's being called these days. Got a short video that was produced for us just a week and a half ago by a very, very good friend, a very long-time friend of mine, almost 45 years, I think, Phil Kornbluth. He had worked for all of the major companies providing helium back in the day. Airco, you might remember the name, turned into BOC, British Oxygen, worked for Linde, Union Carbide, all in executive management, worked for these outfits all over the world, very, very close to the helium industry, and still is. He operates on his own these days under the name of Kornbluth Helium Consulting. We'll start that video in just a minute. I wanted to just maybe take a little informal poll first, and just you know, show of hands, or you know, give a shout out. I think a lot of us are interested to find out what everyone's paying per liter of liquid helium these days. I've personally heard the range of a low of $20 to a high of $100 per liter. And does anyone in the audience want to share what their current typical situation is? I'll hand the microphone. Anyone around would like to comment? Okay. You know, sort of looking at, what is your usage on a monthly basis, or, annual basis, for example? Do you have helium liquefier? Do you rely totally on the liquefaction, your own liquefaction system, or does it augment, or do you augment it with external supply? What are you paying for it? All that good stuff.
Yeah, so I don't have everything at the tip of my fingers, but I actually just was going over the price analysis recently from the stuff I get from my stockroom. So we're currently paying $30 a liter if we're buying liquid helium. Ultra high purity helium gas ends up being equivalent to almost $40 per liter of liquid helium. We luckily do have a helium recovery system we installed about 2 years ago. I've got 5 kind of typical superconducting magnets on it, plus 1 really high use magnet that has to be filled every 2 weeks. So that means with the losses, currently I'm buying 100-liter dewar per year, plus a couple of helium cylinders, maybe another 4 or 5 cylinders of helium gas that I put into the system per year. Was that all the questions? That was most of them.
What is your total usage per year?
Let's see. So now I've got to try and do some quick math in my head. So let's see. So for the facility I manage, that's 3 magnets. That would traditionally have been 400 liters a year, and then we've got 1 other magnet that would be like probably 3 60-liter dewars a year, and then we've got a higher boil-off FTICR horizontal bore magnet that was probably 6 60-liter dewars a year, and then we have this quantum design PPMS magnet that had a recirculator built onto it, that instead of refurbishing it, they stuck it on my system. So now it needs to be filled every 2 weeks with about 40 to 50 liters of heliums. That would have been like 25 60-liter dewars a year. I mean, you wouldn't do that in real life. You'd refurbish the cold head, but yeah, it's a lot of my helium recovery system's running. I mean, it's running solid, like almost 100% of the time, generating liquid.
Good to know. Thank you. Just one follow-up question. Now, your recapture system, is it strictly for fill the bags and you send it out to liquefy, or are you liquefying right on site?
Yeah, I'm liquefying on site. I've got 2 liquid helium transfer dewars, and then the collection bag, medium pressure storage, and a liquefier. It liquefies about 1 liter per hour, roughly.
Good to know. Thank you. And anyone else want to share current experiences?
Yeah, Utah, we have a recovery system that's working quite well, and we have I don't know how many magnets we have, 9, I guess. And we don't recover everything from one of our magnets yet, but it's been working quite well. And I don't know the current price of helium, what we would pay now, because we haven't bought any. So it's been working pretty well, and we're pleased with that, but we're still curious about it. I did not want to buy this system. As an engineer, I said, well, the price of helium is going to go back down. I said, but don't take any of my stock picks either, because I was just dead wrong. I'm very glad we have it installed. Scott and I work together at different locations, and it's been good to, there's been a synergy for this recovery system that's worked pretty well. We're smiling about the recovery system.
I would think indeed you are smiling. Anyone in the room that is just relying strictly on outside vendors? Dave? Please.
So UC Merced, being the smallest university in the UC system, has yet to have a recovery system, but we're working on it. And the NSF has recently put out a proposal call that might actually reach down into so-called low usage that we might have. We use about 700 liters of liquid helium per year to keep a 600 Brooker 600 going, and a Varian Agilent 500 and a 400. And so we pay, well, with tax, my most recent invoice was $3,000 for 100 liters, so $30 a liter, which seems to fit right in. So we're hoping that that the university will see the need to get liquefiers for everyone, given the high price. But we're starting the process this year with the NSF proposal, and then we'll keep agitating for the future.
Thank you, Dave. Good information to know. Anyone else in the room strictly at the mercy of outside vendors?
So I share Dave's, because we are neighbors, Fresno State, just about an hour from UC Merced. We have 3 magnets, a Bruker 300, a Varian 400, and a JUL 600, completely depend on external vendors. I need to choose a nitrogen-cooled probe instead of a helium-cooled probe on our new JUL, considering that helium cost is going to be forbidden for us. So we use about the same amount of helium, about $30, that's what we pay. So our plan is, if things go south further, we might retire one of the magnets to keep the cost down. That's, right now we're able to manage it.
Good to know. Thank you very much. Dave, please go ahead.
Is that I've given some thought to how we might get together, and for one of us to recover and the other liquefy. Yeah, I know. That's kind of a political decision to figure out how it's done, in a bureaucracy. But anyway, I'll give this back.
Thank you, Dave. Is there anyone else who would like to chime in on this rather weighty situation?
Yeah, it's University of Akron. I did the math. We do about 2,000 liters of helium a year because we have no recovery and a very hungry 750. But right now we're at 40 a liter, and we're quoted 45 for the next fill we do. So we're trying to negotiate with other providers who are not taking on new customers for the last couple of years, but we have one that might be willing, and they quoted, I think, 33, which will help us a lot from the 45, but quite a bit different than the other what other people are paying, it looks like.
Thank you very much. Anyone that has had supply limited or shut off altogether? Any particular remarks on it?
Yeah, so yeah, we're currently under, I'm trying to remember the percentage, I think we're at a 40% allotment. I can't remember exactly what the volume is, but in the previous helium shortage several years ago, was this about 4 or 5 years ago, before we got the recovery system, our supplier that we've used for, I don't know, 30 years or however long, they had us cut down to 30 liters a month, and you couldn't save it up between months. So we couldn't get liquid helium from them for our magnet fills, so we had to go to a different supplier, and we were paying, I don't know, maybe twice as much. And again, this was 5 years ago, so it kind of pales in comparison to today's prices. But yeah, and a lot of people are under allotment like that.
You're not kidding, they are. Did you have to shut any machines down?
Yeah, we luckily at that time we were able to get helium from Praxair, so we didn't have to shut any down. But in today's environment, we probably would have had to shut some down.
You're not kidding. Anyone in the room who has had to shut down magnets? Am I missing someone? Oh, sorry, Dave, go ahead.
When the current helium shortage started, the university went under allocation, and in fact, we're in the process of moving one of our magnets now, and fortunately, Bruker has good access to helium, so we were able to proceed. But when we first started, our vendor denied us our PO, or denied our PO, and we had to go through the university bureaucracy for them to assert that we were preferential to get helium. So that's the closest we've come. Hopefully, keep fingers crossed.
It's been challenging at best. Anyone else? Any other comments or remarks? No? You ready to see the video?
Hi, my name's Phil Kornbluth. I am the president of Kornbluth Helium Consulting, LLC, and I'm going to spend the next 15 minutes or so providing a brief update on the status of helium shortage 4.0. I presented on this topic to one of the Ivan meetings last May, so I'm not going to do a complete rehash of that, but I hopefully will provide a decent update. Very briefly, I want to just introduce my consultancy to you. It's a consultancy that is completely focused on the helium business, and I'm a commercial guy, so Kornbluth Helium Consulting is all about various commercial issues related to the global helium business. I've been in this business for a long time, 40 years plus. I've had responsibility for running two of the global helium businesses among, for lengthy periods of time. I've known Jonathan Webb since my days at BOC in the early days of MRI when he was a good customer of mine, and we had a very good relationship, and so we've known each other for, probably since I don't know if it was the late 1980s or the early 1990s, but it's been a while. So anyway, that's who I am. That's what I do. So I'd like to start out just with a very brief recap of helium shortage 4.0. The shortage commenced in January 2022 when the Bureau of Land Management's crude helium enrichment unit commenced a 6-month outage. The primary causes of the shortage were delayed production from a huge new helium plant that Gazprom has constructed in Siberia called the Amur project. The shutdown of the BLM, which took over 10% of supply out of the market. And then throughout last year, there were various planned and unplanned maintenance events. And I'm not going to waste time listing them all because I don't think, the folks on this call really care about, the minutiae of the shortage. There's 4 of the 5 helium majors have declared force majeure and have been allocating supply throughout the shutdown. The only exception to that has been Air Products. Air Products has been very aggressive on price, so they haven't been doing anybody any favors. Contract prices for helium have increased dramatically throughout the shortage, and those price increases have been driven to some extent by very large cost increases that the helium majors received from ExxonMobil beginning of last year and from Qatar mid-year. So they've created a little extra push behind those behind the price increase activity. So where are we now? Well, helium shortage 4.0 is still in place, but clearly not as severe as it was during the first half of 2022. The BLM has operated steadily since last June with the assistance from Merced, who took over operation of the crude helium enrichment unit from the BLM. So we now have a competent operator running the BLM system. There haven't been any major outages in recent months. The electronics industry is in a bit of a slump, so that's reduced demand a bit. But 4 of the 5 majors are still allocating supply, although the allocation percentages have increased to as high as 100% in some cases. Just to be clear, 100% allocation is still an allocation because the customer is being held to some measure of historical demand. They're not allowed to increase demand. There still is no production from Gazprom's Amur project. It's still very difficult to secure new supply. Contract prices are still increasing quite a bit. And the force majeure declarations, which remain in place, combined with the allocations, make it very difficult to obtain quotes on new supply. They make it very difficult to fend off open escalation price adjustments. So what's the outlook for the rest of this year? Well, the most likely scenario, unfortunately, is a continuation of the shortage throughout the year. The key variable is whether production starts up from the Amur project and whether it's able to flow to market, and how quickly production ramps up. All those things. It's all about Amur. The latest forecast from Gazprom is that Amur could restart in June. I would just say there's a very low confidence level in that forecast, as well as anything else that Gazprom tells you. There's also a smaller project in Russia by a company called the Irkutsk Oil Company. They refer to themselves as INK. That's close to starting up. That would add 4% to roughly the global supply. So that's not enough to end the shortage, but it is enough to create, some relief. The severity of the shortage is going to be variable through the remainder of the year, and it will be worse during periods of plant maintenance. So, for instance, the BLM is taking a maintenance shutdown in April for 7 to 10 days. That might impact the market, but ExxonMobil, who operates the largest plant in the world in Wyoming, they have a 29-day scheduled maintenance period starting July 10th. That will be felt, and the shortage will be worse, during that period and after that period while the supply chain recovers. And, last but not least, contract prices are going to continue to move up as long as the shortage is in place. So the big question on everybody's minds, when will helium shortage 4.0 finally end? Well, again, there's a very wide range of potential scenarios that could take place and lots of uncertainty. So the takeaway that I would want you to, what I'd want you to take away from my talk is that it's all about Amur and lots of uncertainty. Really depends on when production from Amur restarts, whether it is able to flow to market, how quickly does it ramp, etc. I do believe it's likely that Amur will restart during the second half of 2023. I couldn't tell you if it's going to be one of the three plants that they plan to operate or two. Two would be nice. In the most optimistic scenario, with Amur restarting, a reasonable ramp up of production, and minimal impact from sanctions, helium shortage 4.0 could be winding down by the end of the year. However, if Amur's restart is delayed, if the production is unstable, or sanctions prevent the helium from getting to the market, the shortage could extend into 2024 and even to 2025 and even all through 2025. So a wide range of outcomes. I want to talk about the war in Ukraine and the impact of sanctions, because that's a big part of what's going to happen with Amur. So right now there's no direct sanctions on Russian helium by either the Russian side or the West. That could change, but for now there aren't any sanctions. The impact of the war and sanctions has definitely delayed the restart of Amur and the startup of INK's project. Reason for that is, the reason Amur is down is they had a fire in October 2021 and an explosion in January 2022, and Gazprom's ability to source replacement parts from Western companies has been, they just haven't been able to do it. The other thing is that Western experts who would help to repair the plant or commission either one of those plants have been unable to travel to Russia. So definitely those delays can be directly attributed to the war in Ukraine. There's been reduced helium production from Algeria because natural gas that normally would have been processed into LNG at the Algerian helium plants has been diverted to the undersea pipelines that flow to Europe to replace the flow of gas from Gazprom to Europe. So Algerian production is down. Sanctions have increased the difficulty of shipping helium containers to and from Vladivostok and just to and from Russia. But Western carriers are not allowed to call on Vladivostok, so it's tougher to get it. There's fewer sailings. It's tougher to get containers in and out. An emerging issue and a very important issue right now is it appears that U.S. manufactured helium containers, which are probably more than 85% of the global fleet, are going to require an export license to go to Russia to pick up helium. 2 early requests for export licenses have been denied, and there's an appeals process going on right now. If Western companies are not allowed to send U.S. manufactured containers to Russia to pick up helium, that will severely throttle the flow of Russian helium into the market and extend the shortage through 2024 and 2025, because the lead times to purchase new containers from non-U.S. manufacturers, and there's only really one, that's Linde, who manufactures in Germany, are 2 years plus. So even though, you know, Chinese companies or Indian companies or Korean companies or maybe Taiwanese companies could still use U.S. manufactured containers to go to Russia because they won't be directly impacted by the export licensing requirement, they just simply don't have the containers to buy a lot of Russian helium, and the time it would take to obtain new containers would be 2 years plus. So that's really the really bad scenario for the shortage, the helium shortage, and it could extend the shortage for several years, you know, at least 2 years. The next really big source coming into the market after Amur is Qatar 4, which is expected to produce 1.5 BCF starting in 2027. So, you know, if you wanted to say, well, if Amur is a total disaster and it never happens, how long could this shortage last? Well, that's your, you know, your absolute doomsday scenario would be 2027. I don't think it's going to be that bad, but again, it's all about Amur, and this container licensing thing really threatens to extend the shortage. So that's all I had. I want to end with a little bit of a plug for Gas World is going to have a helium conference from October 30th to November 1st in Houston. If you're interested in learning more about the helium business, that's going to be an excellent event. All of your suppliers, whoever they are, will be there. It's a great place to network, and if you're not happy with your current supplier, well, maybe you want to meet some of the other ones. But that's all I had. Hope you have a good rest of the day, and I hope this was informative for you. Thank you very much.
So we all know Dan Iverson, and who has been involved with Open VNMRJ, and so we're going to hear a yearly update.
An update.
Yes. No, I think our drawing is after, isn't it?
It's after.
Yeah. Yeah. Right. Oh, yes, right. $500 gift card. So if there are online people who know someone who's not online, start sending text messages.
Don't you think it should be like a new tradition where it's the previous speaker gets the reward?
Well, since he controls the picking.
Hey, that's right, I do.
Yeah, that's right. Yeah, that's right.
Okay, thank you. I'm Dan Iverson. I used to work at Very International as the software engineering manager. I've converted to Ivan here, so I got my brand new Ivan shirt on, and I got my year-old Ivan jacket on, and I got my Ivan knapsack back there. And what I'm thinking, if I had a nice pair of Ivan sweatpants, I'd be a complete Ivan fashion statement. I think that would work out. Anyway, Open VNMRJ. You gave me a defective button here, Dave. Slide switch on the left side. There we go. There we go. So I just wanted to sort of remind people of sort of the timeline for Open VNMRJ. It was originally released in 2016, and it was VNMRJ 4.2, the Agilent with the 110 patch installed. So that was sort of the first release in 2016. Shortly after that, Agilent released me, and then I started to do these things. We put all of the software up on GitHub. It's all open source. Anybody can get it. It's free. We did our next release in 2020, and then another one in 2021. And this crossed out one, so it's been downloaded 4,800 times, which is, I think, pretty good. Last year it was 3,600, and the year before that was 2,400, so about 1,200 per year. That's 100 a month. I'm a bit surprised, but that's a good thing. Just a little update on the current OS situation. There's been some turmoil. CentOS 6 isn't supported anymore. CentOS 8 isn't supported anymore. CentOS 7 will be supported until next year, mid next year. Red Hat has come out with a nice free subscription thing. If you have up to 16 systems, you don't have to pay for the Red Hat support, and that seems to work okay. I would say that the preferred OSs at this point seem to be Red Hat, something called Alma Linux, which is basically uses the same stuff as Red Hat. It's sort of like the old CentOS, but it's now called Alma Unix. And Ubuntu. People seem to like Ubuntu, and that keeps coming along. So, like I said, it was released, this last one was released about a year and a half ago, and since that time we've been, oh, sorry, I missed a slide. So the current 3.1, this is what is supported with the current 3.1. I would call that sort of an OS release. We added all these different versions that it supports. I just heard recently from someone who installed it on the Mac version on Ventura. I think he was the first person that made it work on Ventura, and it also works on the Mac new silicon chips, the M1 and M2 silicon chips. So that's good news. But since we did release 3.1 a year and a half ago, we've made quite a few changes. I think I counted them up. GitHub has a nice little tool that says, every time you make a change, you make a little log file. It's been over 150 changes in the software since 3.1 release. My color coding here, the black ones are sort of bug fixes. I don't expect you to read them all, but if you go back, when we put this up on YouTube, you can scan through them if you want to. It's also on GitHub. It's all in the release notes there. Black ones are bug fixes. The green or blue ones are basically new features. The blue ones I sort of highlighted as relatively larger updates. So like it says, it supports Ubuntu 22 now. So that was a lot of work that Dave helped with to get that working on Ubuntu. And don't touch it. We added all the imaging protocols for Innova systems. The guy who wasn't here, Luke Tremble, that missed out on his prize, he's in Canada somewhere, and he and I have been working to get all the imaging stuff working for on an Innova system. What else I got in there? Some more changes. I should put my glasses on so I can actually read these. Bert Heiss continues to help out. He put in a T1 row experiment, and he also requested that the probe connect mechanism, which people are familiar with if they have a DD2 or DDR system, work on an Innova system, because it makes things set up of probe files and things a lot easier. So we put in the probe connect business. And a lot of the green ones are basically my work with Chris. He keeps asking. In fact, this is out of date. Even earlier this year, he found a bug, which we fixed, and he also requested a new feature, which isn't on the list because it was done like Tuesday, and I had to turn this into Eric before that. Last page, again. So this is just more basically bug fixes and things. A lot have been reported from spin sites. People complaining about various things, which is a good sign. So based on all these changes, I'm planning to support to do a new beta release. probably in June. I'm hoping to keep it to just a couple months. The 2.1 beta release went on for like 8 months, which is too long. The 3.1 beta release was much better, so that went on for a couple weeks, a couple months. I'm hoping this one will be a similar thing. I'm not quite ready yet. Like I say, I have it working on Alma Linux 9, which is their latest thing. But the installation is a little fussy. So one of the things I do is I have to check to see if it's got the right Java version, if it's got Java running. Well, it has Java running, but it's the wrong Java version that they put in by default. It's one that doesn't support graphics. So now I got to figure out if the right Java is in there as opposed to just any old Java. Details like that keep me busy occasionally. So what I'll do when I'm ready, probably sometime in June, is I will email the 3.1 people to see if they're still interested in doing 3.2. And if you weren't part of 3.1 and you want to help out with 3.2, you can email me, and we'll put you on the team to do that. Are we on time? Good. So there's a couple interesting noteworthy posts that came out in Spin Sites, which is our user forum. One was from Jesse, who's sitting right over here. It turns out that Windows 11 has made some nice packages, and you can actually run OpenVNMRJ on Windows now. So what Jesse has done, and you can, this other thing is, the graphics and also some printing, is what he's done is he's made a little installation tool. So you click a couple buttons, you put this on your Windows box, you click a couple buttons, and it will update your Windows so that everything's installed, and it will download OpenVNMRJ and install that, and you're now running on a Windows box, which is pretty cool. I think I might see if I can just put that in the release, but he's got it on his website. I think, at the bottom there, if you want to get it for yourself, he's got it on GitHub on a website that you can download it and use it. The other noteworthy thing from SpinSite was something from Laird, and he found out that if you take a Mac and you install a virtual machine on a virtual box with Linux in it, you can actually run your spectrometer from a Mac, or actually from Windows box. Well, he did it from a Mac, but I assume if it works from a Mac, it'll work from a Windows box. You can just run it directly from a virtual machine with whatever kind of PC you want to use. Okay, so I'm going to finish here with some things that are not OpenVNMRJ. The first is the Ivan webpage at ivannmr.com. And the second is some things that Chris and I have been working on that I will review a little bit. So the Ivan webpage, this is all courtesy of Eric back here. He's sort of our person in charge of everything here. So what you see at the bottom, it says Ivan, Adjunct Spin Sites is now Ivan Spin Sites. And there's two places that you can click. So what happened is when the original Agilent Spin Sites was on a platform called Jive, and when Agilent decided they want to do it anymore, I transported all that to Slack. And then recently Slack changed their policy that says anything older than 90 days, we're just not going to let you see anymore. And since there are a lot of manuals and schematics and all this history that wasn't very useful, there's nothing left on Slack basically. So then what I did was I transferred it to something called Zulip. So the whole forum is now on Zulip. If you were a member of the Slack forum, you're automatically a member of the Zulip forum because I transferred all those accounts. But I didn't transfer the passwords because those are sort of, you don't want to do that. So one of the top one is if you were a member of Slack, you click the top button, put in your email address, and then say, I forgot my password. And it'll send you a note, says, what's your new password? And then you can get into the Zulip version of Ivan. If you were never a member of Slack, click the second one, and then you can join as a fresh new member of the Zulip one. Some other things that are on this page is up at the top, I don't have a little pointer, but the fourth one says past workshops. So all, however many we've done, 40 plus workshops are all available on YouTube. You can go there and view them. You'll probably be able to view this one in a couple weeks. And then the other one that we added was the annual Ivan Founders Award, which is third one from the top. And so, you know, Ron just won it this year. And we decided to make it easier to nominate someone to get one of those fancy globes. And so, again, Eric put this little web form together. All you need to do is put your name, your email, who you want to nominate, and just a couple sentences about why this person deserves to get a nice globe from us. And that's all it takes. The last thing that's not not OpenVNMR, is OpenVNMR.com. So this is our commercial way that Chris and I try to earn some money. So over the last 7 years, he and I have been working on applications that sort of layer on top of OpenVNMRJ. Primarily craft developments, well, totally craft developments. And this is just actually not a full list, but a summary of some of them. He likes his anagrams. So I don't remember what all these stand for, MISQ and CAS. I know there's another one called Aroma, and there's another one called something else. They're basically, you know, over the 7 years, we've built a very good craft toolbox. And now what we're doing is taking the tools and making them into applications that you can use, sort of just, you know, button click applications. So that's the list of most, I guess that's most of them. There might be a few more that I missed. A few more, always coming. He's always changing things. And then as part of this development, when we started this, it was all variant adjuvant. And we soon recognized that, well, our pool of potential customers is going to keep shrinking. And so we said, well, we need to look at JL and Brooker. The first one we did was JL, and they actually have a version, they deliver with their software, a craft version with OpenVNMRJ. And, but that's like, that was how many years? That's 4 years ago? 4 years ago. So it's basically craft version 1, or maybe 2. We're up to craft version 4. And so, but as part of that, we said, okay, well, we need to support all these different vendors. And so what we wrote were import programs to bring in all of these different types of data. And it's actually pretty much separate from craft. It's a separate little package, you know, we call it Invade Pack. See if I know what his acronym is. Don't tell me. Invade is import non-variant adjuvant data easily. Is that right? You basically click a button and you can bring in any of these things. So these are also now just a separate thing. If you just need the data conversion tool, you know, you don't have to get craft, although you'd probably like it if you did. So you can, you know, basically use OpenVNMRJ plus these tools and process any kind of data that you want to do. And I think that's it. Thank you.
So I'll ask any questions.
Dan, thank you very much. Great talk. And hats off to you for continuing to support variant adjuvant NMR spectrometers. You're doing a great job with it all.
Thank you very much.
Couple of questions. Now, is OVJ, OpenVNMRJ, strictly for processing, or are there any components for actual spectrometer control?
It works on, with 3 point, well, all of them actually, they work on any adjuvant variant system. So Mercury, ANOVA, DDR, DDR2.
But what about the actual control of the spectrometer hardware?
Yes,
Okay,
yes,
good.
It'll work as an acquisition system.
Thank you. And next question, sort of two-part. Any feel for how many variant adjuvant systems are still in existence, part 1 and part 2? How long do you think you and Krish, for example, would continue supporting variant machines?
I mean, based on the number of downloads, I get 1,200 downloads a year. That's a bit surprising to me. I don't...
A lot.
But, so it might be just people are downloading for the data processing. I know that some people, like Bert, he downloads it once, but he distributes to all his people in Europe. So in some sense, that count is underestimated. In terms of how many systems are really out there, I mean, I was talking to Jesse's got 6, I know. You got 3, he's got 5, you've got 2. So in this room, we have 15, right? So I don't really know. I would guess, anybody want to make a guess?
Hello. Well, yeah. So I believe that in total, all NMR instruments, there should be, I think, around 9,000 that are active in the world, all manufacturers. Now, take your pick. How many of them have been left? I would say maximum 1,000, probably more towards 500.
I mean, back when we were doing Varian, I mean, we shipped out, 20 systems a month for a number of years. So, I don't know.
Well, the average lifetime of an NMR instrument in a customer lab is around 15 years.
How many years?
15.
15.
Okay, you will find the industry people replace them every 10 years. The academia people will keep them, I don't know, 20, 25 years. So.
Okay.
There have been now 8 years, 9 years that Agilent decided that NMR is not good for science.
2014, they exited.
So yeah, not that many left.
Any other questions? Yes, Scott.
So with that, I'm very interested in that invade thing you're talking about. I'm going to have to look into that. I'm assuming, so when you're pulling in data from, like, they say Brooker, stuff like that, I'm assuming you handle their version of non-uniform sampling and all that sorts of stuff.
Yes.
You essentially double-click on the bit file or a SER file.
So it'll translate some of their parameters like TD into our NT and does all those transfers.
Yeah,
all those
now that I have a mixed vendor lab, that's an issue for some of my users, and I've sort of cobbled together trying to bring like VNMRJ into Brooker, and it's kind of iffy. So yeah, I'm really interested in that.
There's no other additional information you need to provide. You go to the browser. Obviously, this works in the browser, in the open VNMRJ browser, right? You can double click, execute something in the back. Double click the bit file or the SER file. Same thing with Brooker, same thing with Q1 system,
Well, we don't actually convert Mestra. Magritech.
Magritech system is the same thing. You double click on their data on one file.
So you can go to openvnmr.com and request a quote, or you can talk to Krish or I, and we can help you out. Okay?
More questions? Okay.
Oh, one more.
We got one
Dennis.
You don't know how many systems are out there, and we have some too that you probably don't know about.
I knew you had some.
Yeah, 3.1 works great. Could you put something in the system that would ping back to you that says, hey, I have a working spectrometer here? Or is that against the...
I think people...
That's not,
like
not a good
virus?
idea.
I could do that. I don't know if people would appreciate that.
But voluntarily, voluntarily, right?
Okay.
Okay. Well, whatever it is there. Okay, so we're now up for the big moment of the $500 Visa gift card. And so, Dan, get things going there. I'll talk while you work there. Oh, Andy, I'm so sad to say that you were, but you weren't there, the winner of the $500 gift card.
Oh, that's okay. I just stepped out.
Oh,
no
no.
problems.
Oh. Yes. An eel. Oh. Yeah, right? Do you get a special bathroom excuse?
Yes, I actually stepped out to use the men's room. Yeah,
Oh,
but no.
okay then. uh, wait a minute, Victor. Oh, he showed up online, but he's after 30 seconds. Oh boy. I guess we're too close to the restart. I don't know. Yeah. Okay. Okay, so if whoever it was, I forget, comes.
Hey there. I'm actually here.
Yay!
Awesome. Wish I could be
So-
there in person.
Right. Which proves that if you hang around Ivan enough, you're going to win. Okay, so I think Deneen knows what to do to get her prize.
Sounds good. Thanks so much, guys.
Okay. Okay, so I guess we move on. And our next talk is from Travis Greger, and he's going to talk about industrial benchtop NMR.
Is this on? There we go.
A year in review.
There we go. Excellent. Thank you. First of all, thank you so much for the committee for another invite. I don't know if Krish looked at my slides ahead of time. Basically, it has been a year of benchtop NMR, and it really kicked off back in January of 2022, when Paul Boyer invited me to speak along with other benchtop NMR folks at an Ivan meeting. And then, as you can see, some of the topics there for this. And then a year ago, I spoke at the Ivan meeting a little bit more detail than what I'm going to go into today. But really talking about this benchtop NMR in a manufacturing environment. From there, I hosted 2 different benchtop NMR symposium. All right, and then we're going to finish off with this one today. So hopefully it's not the end, but I got to get some more content to really finish this. So, I think, and Krish and I have talked about this quite a bit, but benchtop NMR, it's like beating a drum. You gotta get people to think about it, to use it, especially in the manufacturing setting. They're so comfortable using their near IR probes, or their an XRF or GC for that matter. It's not something, NMR is not something that they think of immediately. And so what I want to do is really go through a series of examples where it's been a journey for myself, for my colleague Chris, who spoke right there, number 4. He's down at a factory down south for 3M. And another colleague, Eric Perkins, who was a contract worker, and we hired a bunch of contract workers who are now at 3M, so there's a very good chance that he might be here someday talking about benchtop NMR. So as I was saying, we're going to be talking about benchtop NMR at 3 different locations. Hopefully, I know the AFE has been approved. I don't know if it's been actually bought yet. So hopefully we'll have some more very specific examples on that in the near future. So, quick show of hands, who's seen me talk about the benchtop NMR talk from last year? I know I see some familiar faces, all right? So there's enough people who haven't seen it, so forgive me if I reproduce this. At one of our factories, 3M is very good at coding solutions on a web, all right? What they're not at the time good at is figuring out if that coding solution is actually going to work, all right? And in this case, there was a particular product where we were coding this solution on a web, and the only way they knew if it was going to work is if it was going to wind up and just keep winding. If not, it was going to wind up and just crash. All right, so they would have to ultimately, run it, hope it works. If not, they're going to scrap that entire bundle of polymer. So Tony Bush, a colleague of mine at the factory, he wanted to buy an NMR because he tried so many different analytical techniques to figure out how to characterize this coding solution in order to make sure that it was going to code correctly. And it turns out that this coding solution had two components, and we're going to just call them A and B. And it turns out that for IR, it was very good at detecting component A. XRF was very good at component B, but you couldn't get it to work together. All right. So what he had to do is he had to figure out a way of doing that. And he's like, hey, Travis, let's do this by NMR. And so we spent a ton of time trying to figure out the nuances. And I talked him off the edge of buying a 400 megahertz NMR because, as we all know, it's not just the instrument that you need someone to use it and operate it and maintain it. And I think if anyone's familiar with situations in factories, that is very, very difficult to do, to justify having a person work full-time on a piece of equipment in support of a product. So what we did at the time, Tony and I worked quite a bit with Paul, actually, when he was at Magratech. This is a 60 megahertz NMR of that coding solution, where you can see our solvent peaks, and then down below you can see components A and B, where we've got decent resolution. I mean, we were able to get some very good integration. In comparison, at 500 megahertz, nothing beats field. Nothing beats field. I think you're going to see some examples later where field is good. And so, we were trying to figure out best way of integrating these peaks, making sure they're resolved. You can see blowing up of that aliphatic region, component A, had a bit of a shoulder that, we were concerned with whether or not that integral area was going to impact that ratio. And ultimately, we wanted a ratio between A and B, and we had a range that our target was. Very easily at 500 megahertz, we could see it. So what we started to do is compare between those instruments. We made multiple samples that were able to demonstrate these different attributes for qualifying this instrument. Now, the first instrument I qualified was a GMP facility. Now, 3M only has maybe one or two GMP facilities in the entire company. Unlike many of the pharmaceutical companies here, companies here, where they're almost all GMP. I installed this instrument early on, and we demonstrated, we were running it. And if you want to see more of the bigger story, you can go to the Ivan NMR website and check it out. But what we were very interested in was trying to develop this piece of equipment that just anybody could use, push button operation, and have it qualified in a way that if the instrument was ever or the factory was ever vetted by the audit authorities, I guess I can't think of who the audit authorities would be at the moment, to show that we did due diligence, because there wasn't a whole lot of benchtop NMR qualifications at the time to audit and show that the instruments are working correctly. And so we spent a lot of time going through and demonstrating these 6 different attributes for, to demonstrate the instrument is working correctly and every time, and that anybody could use the instrument as best and as uniformly as possible. So, what we were doing was taking it, and they haven't had to scrap a a roll of material ever since, but ultimately, the benchtop NMR worked was exceptionally successful. They bought a second instrument, which is, I guess, a requirement in a GMP facility, and now I understand why, because the power supply went out on the instrument, which was almost unheard of, but they're back up and running, but this is why they do that. So, as I mentioned before, that 5-ish minutes, in a nutshell, was a previous talk from last year to talk about all the details, the nuances, the struggles to qualify that instrument in the manufacturing environment. What we're going to do here is look forward a little bit more and look at some of the new applications since then. So, talking about this, the second instrument that went in at 3M, this came directly out of one of the slides for the purchase justification. We ran quite a few samples for many, many years for Chris. He'd ship them up. We'd get to them as soon as we can. All right. Even though, all of us knew Chris, he worked in St. Paul for quite some time and then moved out of state to the other 3M location. He really wanted to be able to start using this piece of equipment on site. His background was ICP, so inductively coupled plasma, which measures, part per billion levels or lower of metals. All right, so he's not an organic chemist, but he still wanted, an access to an NMR to solve problems almost real time or near real time, so he wouldn't have to ship them up. So, and as I mentioned earlier, if you go back and watch Chris's talk in one of our other roundtables, he talks about that journey, the thought process, the struggles to learn NMR from a beginner point of view. So, what we have here is 3 different options where, option 1, shipping the samples to us, we'll get to it. Option 2 is purchasing the benchtop NMR, and option 3, purchase the high field instrument. And ultimately, this was a very good decision on his part and management. We've got to give management its credit to buy the instrument, and clearly the instrument has already paid for itself many times over in the short time that he has had it. So what we're going to go do now is go through a bunch of examples of that Chris worked on along with myself and Eric to show the utility of this piece of equipment. You look at those broad lines, all right? You look at these C-13 satellites. 3M is a polymer company. We look at a lot of polymers. Coming from grad school where I did carbohydrate chemistry, where you saw a lot of coupling constants and beautiful NMR spectrum, coming in and looking at these broad lines of polymers, even at 500 megahertz. It was a little bit unnerving initially. Now that I'm at low field or benchtop NMR again, it's like old school again, and part of the things that we're struggling with right now, a little sidebar here, is remembering how insensitive these instruments are. We got spoiled. When I was hired, sorry, Ron, when I was hired, we had three varying systems, and 1 solid state, and they all had Nalorac probes, I believe, at the time. And, where you'd run a single HMBC overnight. And that's just the way it was for the first few years, and then the cold probe technology started coming out, and you've got more signal to noise, more data than you know what to do with. Now with the benchtop, the one instrument that we have access to does not have a sample changer, and it's across campus. So having a sample changer with a benchtop is an absolute must. So you can just it's just time, just collect more data. And we're not collecting 2Ds per se, but the more data you can get, the better. The demonstration here is we had these 6 different samples with various levels of this additive that really impacts performance of this polyester. all right? And, you know, to be able to pick and choose different lots of material for ultimate performance was key. And NMR was the absolute best way of doing this. So we were able to clearly demonstrate, you know, that we could see down to roughly 200 ppm of polymer in this polyester. We were able to take it a step further, and the team has been working quite a bit with Kraft lately, and, you know, integrating that shoulder peak accurately can be quite difficult. So, you know, being able to isolate that peak by itself, having baseline resolution around the peak, makes things that much easier. And so we're going through the process of optimizing and teaching other folks in the department how to use this to, you know, accurately measure, because even at 200 ppm, that impacts performance of our materials, surprisingly so. So you know, anything that we can do to help the manufacturing folks run as efficiently as possible, anything we can do to help them is just that much better for everybody. So this is another example here. Make sure I keep an eye on my time, where resolution's everything, or field is everything. You know, at 600 megahertz, we have this other polyester peak where, you know, we can see all this very, very nice baseline resolution. You know, here in the polyol region, you know, we can see all these nuances and individual species of diols. But, you know, at the lower field, you know, we can kind of, well, maybe we can just integrate this whole area as opposed to individual peaks. And then again, you know, maybe we can integrate through here, maybe through here. And then we've also got this one, this peak here, where we can just integrate this area. You know, but, you know, how do we get some very good measurements? Because what we want to do in this case, we were trying to qualify a second source. And there are some other, these little components down here actually are really important to the performance, again, of our material. They control your, I can't remember if it's this exact example, but you know, your crystallization temperatures, your TG, your rheological properties are all impacted by these low-level additives, which is not something necessarily you think about on the NMR side of things. So, you know, again, you know, we're comparing at 660 megahertz, the differences between all these different major components. So it's 4 major components. And, you know, at high, you know, for the larger components, these actually, you know, between the yellow and the green, pretty decent agreement. All right, even going from, you know, those two NMR spec, you know, between the two NMR. Where we start to get into trouble is this blue area right here, where I was subtracting, you know, integral areas to ultimately get those low-level values. All right, and, you know, I think in the long term, even though they don't agree with each other, we've convinced ourselves that as long as we follow the same procedure every single time, you know, we'll start to see slight differences in the NMR values, which will, you know, suggest equivalency or not. So that's really important. We've got a high field instrument, high field data here of another polyester, and you can see these peaks right here where the intensities are actually rather important. You can also see the shoulder right here that is rather, that's what it is. So this is at 600, and this is at 60 megahertz, where you can see these shoulders, you see the trends are the same, all right, but you just don't get the same kind of baseline resolution you do at high field. All right, so it's, you know, it's going back to just kind of old school mentality. You know, what can I do with the data that I have? Again, you know, comparing between the instruments, the baseline resolution between everything, and calculating. Now, this is where it gets really interesting, as I mentioned earlier. You know, talked about this component A as a function of TG. All right, it is a pretty darn good correlation of, you know, collecting that data at 60 megahertz and showing that, you know, if we can accurately measure or even get a trend of that component A, we can start to predict the performance and the overall TG, along with your crystallization temperature here of this other component B. All right, not so much on crystallization, not so linear yet, but if we had maybe some more resolution, we might be able to take a step in that direction. So very nice correlation that demonstrates how we're correlating the NMR data to the thermal properties. Another one here. All right, so here again, different polyester. So in this case, we've got it at 80 megahertz. You can see these trends on the side, and just qualifying these materials to show that if we're trying to find a second source or a is this material good and bad, the benchtop NMR is actually working very, very well. All right, even though we don't have baseline resolution, we can even see differences in additives that may or may not supposed to be there, and it can be all integrated very nicely. In this case, here's a raw material which at a high level looks rather good. All right, all your major components are there, but when you start looking at the baseline, you can see these small materials here that were not supposed to be there in the first place. We had about 6 or 7 samples of this raw material that were shipped in from another 3M factory to Chris, where he prepped them, just collected the data, and for every sample that had these extra peaks, correlated to product that was not made correctly in the end. So, just another example of the utility of using the NMR that was not able to, the NMR was used where other analytical techniques failed. All right. Again, a little blow up there. So in this case, we're dealing with different isomer differences. Lots of cis and trans isomers, which ultimately correspond to different performances in crystallization of polymers, in this case, a urethane. So they're able to head off any issues with the manufacturing process by looking at the the NMR data. In this case, I don't think any other analytical technique would have worked. NMR is the only way that this would have been done by looking at the different isomers. Very practical example. All right, they were starting to get some new additives for this. I think this is another polyester, where our supplier had typically shipped us these little, egg-shaped, pellets, polymer pellets. And in this next lot of material that came in, we saw a few of these cylinders. Just they weren't what they were supposed to be. And if you took an NMR of the material, we had this extra spike, which if anything about 1.2 ppm, I know what that is. All you have to do is just see if we can correlate it. And sure enough, year round, we're missing that peak, all right, right here. And you see it right here. All right, so 1.2 and about 29 ppm is a long-chain hydrocarbon. Call up the manufacturer. Oh, yeah, we had a leak of a lubricant into your lot of material, which just would have just done bad things to our lines and would have shut us down and everything. So, a very practical reason of having the NMR spectrometer at the location in the factory going for there. This is actually a very interesting. We were consulted. Chris collected this data, and we had some water tank rinse. And there's an additive that we're not going to go into, but he collected some very nice data where it clearly demonstrated that we had an additive in there, and he needed to know how much it is. But because you had such a shoulder on this, how are we going to accurately integrate this? All right, and so I'm just showing you the 80 megahertz data. We also supplemented with the 600, but we used his data, and this is down to about 0.2% of this additive in water. And this gets back to my comment earlier, at 600 megahertz, we probably saw this in, maybe 16 scans on a cold probe. All right, this was probably, 500 scans or more. But with that sample changer, he sets out and walks away and just keeps the whole thing running continuously. Whereas, on the 3M campus, we have to walk to another building to change samples out, which just takes, gets our steps in for the day, I guess. But just a very nice, because the factory needed to know how high this level was in order to fill the paperwork correctly prior to disposal. So just another example of because that additive does not GC, I can tell you that right now. So NMR is really the only way of doing it. So 2 more examples in the last few minutes that I have. So and this is hopefully a new instrument in another factory where we have these oils and we use them as part of our process, but over time they age. All right, and we've got about 8 to 10 different oils that if you look at them over time, you see how they going from new to old. All right, and so what we're trying to do is correlate this. How can we predict the lifetime of that oil so we're just not automatically changing it too early or too late when the performance of that final product is not where it needs to be. So what we're starting to do is take the products from these different lots of oil, or when those oils samples were taken from this one lot of oil, and correlating that to ultimate product performance. So that's where the utility, again, at this new factory is going to be really powerful. I don't know if I showed this one or not. So this was another same factory where, so this is an adhesive at 500 megahertz, where the adhesive was tackified with this new tackifier, and in order to stabilize that tackifier, we had to add these additives. All right, because it sits out in the sun, it oxidizes, cross-links, and then it doesn't stick anymore. And so, at 500 megahertz, clearly see, we take this adhesive, dissolve it up, easily seen. All right, but those same samples, not very obvious. All right, here, we got 4,000 scans. Takes a long time to walk back and forth, not very practical. You might be able to argue you could get some bumps in the baseline, but just not very accurate. So this gets back to just more of the practical side. What can I do to the NMR sample to make sure that we get decent signal to noise? What I wound up doing was prepping the sample. Again, we went from 4,000 scans here to 256 scans here. Very reasonable. What I did is I just prepped the sample as almost a plug of jelly is what it looked like. And so, it's already polymer to begin with. The lines can't get that much broader, so, even at the low fields that we're at. And we can clearly see the additives that are supposed to be there. We need to make sure that they're present there at a specific level, because for warranty purposes, we just want to make sure it's there so that if there's an issue, we can point to a root cause. So what I wanted to do today is just show you some applications, really beat the drum. I think the confidence level of Chris Thomas, because I think he gave that talk over the summer, something like that. The confidence level of Chris is really going up when it comes to using this spectrometer at the factory. They recently hired an application engineer at the factory whose background is PhD organic chemist. He heard that Chris had a benchtop NMR, so he's going to use the NMR. They're starting to use it everywhere to solve problems. All right, and that's what it is. It's another tool in the toolbox to solve problems where they haven't been able to solve before. And part of our mission in St. Paul is to help him in any ways possible, whether it's software, automation, or just a little bit of confidence to make sure things are going the way they're supposed to be. So with that, I just want to thank everyone, especially the folks at 3M and the Ivan, and then the folks at Magritech. So, and thank you for your attention. So, any questions? Anything online? All right. Thank you. I think, I don't need this. I think with the benchtop, you just get a little bit more getting your hands dirty. With the low signal to noise, you got to get your hands dirty and figure out new ways of doing things. And you don't have signal to noise to burn or or enrichment like we did like from the earlier talks today. So it's good. But thank you.
Okay. So, thank you, Travis. So our next prize. What? I'm sorry? Oh, okay, yeah. Our next prize is a $50 Visa gift card. Rui Feng. Ah. Oh. Uh-oh. Paul Oblad. Okay. George Gray. Okay. George.
I'm here.
Yay!
I never expected that.
Congratulations, George.
Well.
And you'll be in touch.
Yeah, I'll
Okay.
be coming down Monday, so I may be able to see you.
Okay, that's great. Our next talk is from Don Bouchard, representing Q1, and he's going to spend some time telling us more about Q1.
Great. Thank you very much, Dave. Can you hear me?
Yes.
Okay, good. Well, you've all heard my spiels at all of the Ivan meetings, so a lot of this is going to be a little bit of rehash. What we're really doing is one system at a time, because we're a small player. We're not promising anything in terms of a cryoprobe, but we are making a very economical, very rugged console upgrade plan from 300 to 500 megahertz. We offer 600 as well, but we're still developing the 600 magnet in the factory. But we are making 400s in quantity, and we're making a longer hold time doer than the typical AS400. The hold time is now up to about 240 days, which is an improvement over 180 days. It's a little bit bigger doer. But the right now we have, going the wrong way. We have 5 Quantum 1 Pluses here in the US. 3 of them are at commercial sites. One is our demo lab, and another is at the University of Mary Washington, which is our longest install now. We haven't had one hardware-related failure in the last 3 years, so that's a promising indicator. The for a upgrade package, we can do upgrades in a couple different flavors. We can do a very basic upgrade using a customer's existing shims, probe, and whatever else, shim stack, with a Quantum 1 Plus console for $150K. And we're currently waiting to deliver a system to a college in Baltimore, where we're just reusing their Brooker probe and Brooker shims and integrating it with the Quantum 1 Plus. For about $60K more, we can go full automation with a Q1 STM probe, Smart Tune and Match, the 24 or 60 position auto sampler, and the Q1 Quantum 1 Plus console and auto sampler and a new computer. So it has advantages over an older system where if you're just buying, say, a used system, because you're getting a brand new computer and a brand new spectrometer with a warranty and a reasonable expectation of the lifetime. So I like to say it's excellent performance at an unbeatable price. And it's a compact system. In the top rack is all the RF generation. Middle rack is the power amplifier, 3-channel deuterium, proton, and X channel. And then the lower rack is the shim power supply and pneumatic control. So we all know that the probes are the key to good improving performance in any system, and we offer the hybrid tuning mechanism for faster tuning and unmatched reliability. And this is a patented design, free from drift and hysteresis, with good signal and noise and good solvent suppression. And it's made by our Q1 team in Zurich, Switzerland. So the configurations can vary from just an HX and HF probe, broadband detect, or indirect detect. And then we also offer fluorine tuning on both X and F channels as well. That platform is also available for older Varian and Brooker systems. And so the scheme is to integrate directly with Topspin and VNMRJ just by putting oh, that doesn't show up there. The unit on the lower left is just the Q-Link control, and then the box on the right is integrates with the existing preamp for the tuning control. And then we've also made custom probes with, say, 4 channels at 400 megahertz. This system was at BMS for a few years. And we've also made custom probes, a low silicon background probe at 500 megahertz for our Brooker Neo system. And the performance was equivalent to the Brooker probe without the probe background. We're introducing an inexpensive VT accessory for VT air down to -20 C. And it's priced around $8,000. So if you're looking at upgrading your VT unit. But what I'd like to just spend most of the time is talking about the software platform, Spin Studio J, and how we're adding on every release. We have about 2 releases a year. How we're adding all kinds of features in every release. So it's a modular plug-in system, so if you have a need for either acquisition processing or post-processing needs, it's just a Java plugin. The Spin Studio J, it's a visual or XML programming language with a visual display of the programming result. And we've got a fairly large library of pulse sequences for 1D experiments, X-nucleus experiments, selective experiments, solvent suppression, instrument tests, calibration sequences, and all of all the popular 2D experiments and different variations. HS experiments as well as XH experiments, J spectra. So a lot of these plugins are all built in Java for capabilities like smart tuning and matching. All the automation for both multi-user security. We have a web interface for remote sample submission as well as following the progress of the experiments on a smartphone. 1D and 3D gradient shimming for easy use. And a shape pulse experiments or shape pulse kit for multiple solvent suppression as well as selective excitation. And so I think I'm going to close here. What we're looking for is people that are looking for economical upgrades in a robust package. We know we're probably not going to pass the test of a lot of faculty members demanding, that they get the best system possible. But what we will provide is the same service and support for not a lot of money. And but you still get good performance that's comparable or improving on systems that have been in the field for 10 or 15 years. So I'll close here and answer any questions.
Oh. Okay, thank you. So questions?
Yeah, I just have a question about the operating system that your software runs on.
Windows and Linux.
Which flavors of Linux?
I think right now it's Red Hat Enterprise Linux.
Okay, thank you.
You have a question? Oh, okay. So thank you, Don.
All right, thank you.
Okay. So our next prize, we're up to a $100 gift card. I have a good feeling. Bob Burno. Okay. Chad. Yay! Come on, Chad. Somebody text message him. Chad. Come back to your computer. Three seconds. 2, 1. Oh. Ken. Oh, okay. If he's not online, might as well. Yeah. Richard Rustandi. James Ramini. Okay. Yay!
Am I the last name?
Okay, so you win the $100 gift card, and Shelley will be in touch.
Thanks so much.
Okay, so where am I here? So our next talk will be online, I believe. Is that correct? Yep, Jackie Thomas. And her talk is going to be craft-enabled fluorine-19 analysis.
All right. Well, first off, thank you for this opportunity to talk today. I really appreciate the committee inviting me to give a little snapshot into some of the NMR work that happens in the consumer goods industry. And so, yes, today I'm going to be talking about craft-enabled fluorine analysis that I did in partnership with some of my oral care analytical partners, Poulhazan and Merced, here at Procter & Gamble. And I apologize, I am using the PDF version of my talk because I had to use my personal computer. P&G doesn't like Zoom and all of this stuff. So, so first off, for those that don't know me, I know some of the NMR community, but for those that don't know me, my name is Jackie Thomas. I'm originally from Texas. I went to Texas A&M at College Station, and I have a degree in organic chemistry, physical organic chemistry. I worked for Dan Singleton, and we did a lot of C13 kinetic isotope effects at natural abundance. So I got really familiar with NMR in graduate school. I love NMR, and found my way back to NMR. I joined P&G in 2008, and I worked in upstream technology. I worked a lot with suppliers, raw materials, process lines. And then 2016, their analytical NMR group had an opening, and I jumped back in headfirst, and I've been loving it ever since. I'm also very active with ACS. You'll see me at the local Cincinnati section, national sometimes, and then I'm also an ACS career consultant for any of the students that are attending that are interested in thinking about careers in industry. So a little bit about P&G. Hopefully most of the audience knows, maybe not the parent company, Procter & Gamble, but a lot of our brands. So we are a company of leading brands to help consumers make their lives easier cleaning. So we're into surfactant technology. So we have Gain, Tide, Dawn, Mr. Clean, Safeguard, Swiffer, Charmin, Always. But one of the products that I'm going to be talking about today for this particular talk is about Crest. So Crest is one of our dentifrice products, also AKA toothpaste, right? And so one of the important pieces about Crest is that we deliver a fluoride treatment to consumers to impart the benefit that they are really looking for, whether that's whiteness, cavities, gingivitis. And so we have different product lines, and they all contain different sources of fluoride. Some are formulated with sodium fluoride. Others are formulated with stannous fluoride. And then the one in particular for today is formulated with sodium monofluorophosphate, which is really important because if we say we're going to deliver a certain amount of fluoride to consumers, we really should be doing that. But we're also, because Crest is regulated, by the FDA, you know, it's really important that we are indeed delivering that amount of fluoride. And so sodium monofluorophosphate is really the molecule today. It's a very small, simple molecule. You can see it's a phosphate group with a fluoride attached, and there's 2 sodiums to balance the charge. Lots of consumer goods companies use it in their toothpastes. So, we also call it MFP. So for the rest of the talk, instead of saying the long name, I'll just say MFP. Traditionally, MFP is characterized using ion chromatography methods. So they have a reference standard, usually that's pretty stable. They store it under stable conditions. And then the sample of interest is then compared against it. And so its relative intensities are then compared to give you the concentration. But the problem occurs is when if there's an issue with the reference standard, maybe the reference standard wasn't held at the right temperature, storage conditions, there was a decomposition that people weren't aware of. And so now that reference standard can come into question. And it did most recently within the last 6 months. So there was suspect that there was a degradation to the reference standard. And because of that, the results of the testing for our particular lot of MFP that was going to be made into, you know, hundreds of thousands of products it came back much higher than expected. And so people were asking, this doesn't seem right. This is weird. It's definitely off. Quo, actually, my partner from Oral Care Analytical came to me and was like, hey, Jackie, we have this issue. The LC method is coming back way too high. We don't know if it's actually decomposing, if there's really MFP there, because the ion chromatography method doesn't give you structural information. And so he had found this paper through a brief search, and it was published, and it was actually an extraction method. So people were extracting out sodium fluoride and MFP from a formulated toothpaste, and then doing fluorine NMR on it in a quantitative way, and comparing and contrasting the quantitative method of using a calibration curve versus using an internal reference standard. And so we hadn't actually tried this in our labs. So Quo was kind of like, can you do this? We're on a really tight deadline. All this product is you know, we really needed to really understand the level of MFP that we have in this product. So sure, we could do it. They had a really nice outline of the sample preparation, the internal standard they used. They made the assignments. They talked about, you know, all of the different parameters to set up the experiment. So we were able to quickly, you know, do this and reapply it and analyze the MFP in our lab. And so you can see here, MFP, it's split into 2. It's a doublet because of the coupling from the phosphorus group. We have our internal standard, which we copied directly from the paper, sodium trifluoroacetic acid. I put sodium trifluoroacetic acid. Should be sodium trifluoroacetate. So we were using the acetate, the acetic acid. I don't know if you've ever used trifluoroacetic acid, but it's very volatile. It can evaporate. And then you have issues with quantitation. So no, we were using sodium trifluoroacetate. Sorry about that. And then you can see the free fluoride as well. So the spectra looks good. Everything is great, right? Until you start increasing your baseline. And so as you increase your baseline, you can see rolling effect. And then if we zoom out to our entire spectral window, you can see this horrendous background that's coming through. And so what we use right now is we use a Brooker system equipped with a cryo prodigy probe. This is off of our 600 instrument. And so we have this rolling baseline, and we also have obviously contributions to our background from the Teflon components that are made up of this probe. And so there were a couple of different strategies if we ever wanted to continue to do quantitative fluorine, as we could buy a fluorine specific probe. We don't want to do that because we really don't want to move in and out these Prodigy probes. I don't know if you've ever had to try to change one out. It's kind of pain squared. It's not easy to do. So we like keeping our Prodigy probe in at all times. We can apply strong baseline corrections and do phasing. And that's all well and good. However, as probably a lot of people in this audience know, that it can be biased, right? So if you have a newer person doing the analysis versus somebody who has a lot of experience, phasing can definitely influence sometimes the values that you get. We've also been playing around with craft software. So we did purchase a license in past years for craft, and we were using it to separate minor peaks from polymer peaks. And during the training, one of the things that always stuck with me about craft was about the time domain processing. And so this is just a summary slide. You guys are all very familiar with craft, but when I use this presentation internally, everybody's kind of like, craft? What's craft? And so just to quickly summarize, time domain NMR signal processing is really what we're doing with Krish's software. Separate out overlapping signals from ingredients and mixtures to successfully quantitate those ingredients. And like I said, one of the big things from the training with Krish, whenever we did purchase this, was, he talked about eliminating the issues that arise from baseline and phase. And so if we think about traditional FT analysis, right, you have your FID, you Fourier transform it, and now you have phase and baseline corrections as parameters because those are as a result of the Fourier transform. If you're doing the analysis in the time domain, right, the baseline and phase don't even exist yet. And so it's really the amplitude and, whether you choose to phase your resulting FID, it's up to you. It's really, you can rebuild it and visualize it and use it as a visualizing tool. So that's one of the things where I was kind of like, oh, well, interesting. As I thought about some of the applications we use in fluorine analysis and aluminum acquisitions, we always have these contributions from the backgrounds, from the internals of the probe. So this was a great opportunity to use this software in a applied way for us. And so you can see here is my the FID that's separated using craft for the sample that I was looking at, the MFP, the internal standard. Here's it's reconstructed, and here's the craft output table. And you can see here are my frequencies, and I'm able to assign them, copy down my amplitude. Since I used an internal standard, we have that as well. And, as if you blow it up, those baseline contributions are not there because I'm really just looking at that one frequency itself. And so just like with integration, you can swap out amplitude for the integration. And so, knowing the amount of my internal standard, using my sample weights, doing the equivalence of fluorine, I'm able to calculate the concentration of my my MFP. And I just put this in here just to kind of highlight, if we look at the distribution of 4 points, four replicates. So we prepped it 4 times, ran it 4 times, and did the craft or the analysis on them, right? We can do lots of different types of processing. We can do integrations, we can do baseline corrections, and I just kind of did a couple of them. Some of them come really close, to having a tight distribution of the results. Some of them, if you're not careful, especially if you have, people who are newer, that don't really understand some of the baseline correction or phasing, it can cause lots of issues and lots of variation within the result that you get. And so why not just do a Whittaker baseline correction and then do some phase? When you do that, you get a pretty nice flat baseline. But again, if we blow it up and we just do 1 or 2 clicks of phase to the right or to the left, that could be about 1% difference of MFP. And that's a huge impact to our formulated product, because I don't know if you saw, the level of MFP that we're talking about putting into this product is extremely low. So that could be a big factor in whether or not we're meeting the right target range to deliver the product that we say that we're delivering. So in a nutshell, here are the results. We were able to look at an older retained lot of our standard. We have a new standard that's here. So our old standard should have been about 97. And as you can see, it definitely had decomposed and now is like 93. So we have a new standard. We've been holding it in stability. We will be monitoring it using this NMR method over the next couple of years, maybe a year. I forget how much. They say everything in months, and it was just a lot of months to me. So yeah, everybody was pretty happy with the analysis. We know it's MFP. We have a pretty good, robust method. Doesn't take us a lot of time, quick 10, 20-minute acquisition because we're doing it quantitative. And then, the analysis is not very long at all. So with that, that's kind of what I had to share today, practical application for this audience. And I'd just like to acknowledge the NMR MRI team we have at P&G, the corporate functions analytical section, the oral analytical team that brought this problem to me to kind of work out, internal funding, the Ivan NMR planning committee. To the audience, thank you. Really appreciate it. Wish could have been there, but had to do this virtually. So yeah, if anybody has any questions, I'd be more than willing to take questions.
Okay, thank you. Questions? Yes.
Thank you. That's an excellent presentation. My question is, the NMR spectra that you showed, is it the toothpaste itself or just the pure compound?
Yeah, so the spectra that I showed is just the pure raw material MFP itself. The paper did cite that they do extractions of the toothpaste to recover the MFP. I think they pretty much just make a slurry. And so we've actually been starting to do that as well, because after products are made, that's one of the things that we have to look for was it made as intended? And so they've done other types of titration methods that give them indirect evidence. So they were actually kind of interested in this application, having demonstrated it on the raw material.
More questions? I guess. Okay, well, thank you.
All right.
Okay, so our next, what are we up to? Up to a cutting board. So for those that weren't here this morning, it's a beautiful Ivan cutting board, and which actually can be used as a cutting board, though I wouldn't want to. And so let's do our and Mirich? Ana Sikos? Okay. Prashansa Agarwal?
I am here.
Yay! Okay, so you win the cutting board, and of course, get in touch with Shelley.
Sounds good. Yep.
Okay, congratulations. Okay, so we're up to Krish, and Krish Krishnamurthy, and he's going to talk about time domain analysis, back to basics.
Thanks, everyone. Here I am, back again, between you and the reception. Let's see how long I can hold you here.
Well, you have a little extra time, but there presumably are cocktail or wine and beer or whatever waiting.
I know. It always happens every year, so that's okay. That's it's good to see that there are others who probably hinted about craft. But I'm not really going to talk about craft per se today, even though I will interchange time domain analysis with craft, because somebody told me that craft is a good software, so I have been using that. But fundamentally, what I'm going to talk about is generally applicable to any time domain analysis that you might be doing. Time domain analysis of NMR data. So let's first put some basic definitions out of the way. I introduced a new word there. Frank Delaglio was very excited about it. It's a tabular domain. So we all know about time domain. We all know about frequency domain. And I said, okay, let's have a tabular domain. The time domain is FID. It's a linear sum of frequency, amplitude, decay rate, constant, and phase. A linear sum of a variety of sinusoids. Tabular domain, you think of it as the same 4 information per row in a spreadsheet. Yeah. A frequency domain, we all know. Fourier transform is XY representation. If it's a 1D, XY representation of some kind of a height point against frequency. Yeah. So that's one definition out of the way. Next definition in my title was back to basics. So I want to talk about basics. I'm really going back to basics. Yeah. Signal to noise, line shape. We all measure line shape. I just picked it out of Google. Yeah, I just Googled it, and it came out of, these are the pictures that came out. I put it in. Resolution. I don't know how many of you have done resolution measurement with ODCB. This is really, there's probably very few, probably this audience. When I was in USC, George Ola's lab, we actually used to have the running winner of how somebody can split this peak in ODCB, and whoever gets it done for that week, their name goes up, and so on and so forth. And then obviously, finally, there is this, the newer one that a lot of people use now in testing probes and things like that is water suppression sucrose. You measure the residual line shape of the water. So it measures all three in some form or other. So that's the basics. That's exactly what I'm planning. I'm hoping to talk about, but from the perspective of time domain analysis. So time and frequency domain. So when we want to talk about these basics in time and frequency domain, we need to understand what the properties are in time and frequency domain. Yeah. So in time domain, it is frequency, exponential decay, amplitude, phase, and of course the ubiquitous noise. Yeah. In frequency domain, it's the same four. Same four parameters, just different nomenclature, different way they are expressed. And you also have noise in there. It's very convenient in this particular representation. I put the signal as a nice sinusoidal, decaying sinusoidal function, and I put the noise down there as noise. But in reality, what you are looking at is noise is on top of your signal. Yeah? Noise is not some foreign material. It is a uncertainty in the signal measurement at every point. So it's a part and part of your signal, really. So when you think about signal to noise, it becomes slightly different way of looking at it. So in signal to noise in frequency domain, we all know this, we do this all the time. We either measure the peak height or some kind of an integral, and then we measure an RMS noise, and then say, okay, this is my signal to noise ratio. In time domain, that's interesting. Signal to noise changes as your signal continues to get detected. The signal to noise in the very front of the FID is very high. Signal to noise in the end of the FID is very low. Yeah? That's fundamentally, there is a difference in the definition of signal to noise in time domain versus frequency domain. I keep putting that noise definition there. It is an uncertainty in signal measurement. Right? It's not something that is floating around. So how do we, so interestingly enough, in time domain, if you focus on the very front end of the FID, you are dealing with a very high signal to noise. Right? Whereas in frequency domain, it doesn't matter because the noise got spread out. Irrespective of where you measure, irrespective of which signal you measure, the noise is noise. Yeah? There's a subtle but interesting differentiation between the two. Frequency separation permitting, meaning that if you don't have to acquire long enough, why acquire too long in time? If you're going to do time domain analysis, you can actually have a better signal to noise. Okay. So let's see if that hypothesis is good. So here is a simulated, I don't have a spectrometer. I can't put one in my garage because I had to move my car. So I don't have a spectrometer. I just do synthesis. So here is a craft analysis, time domain analysis of a singlet, a single peak, a signal, and I just add a random noise into it. Right? I do the analysis so the signal gets separated out in time domain and the leftover is residual. That's my noise. Yeah? So I want to compare the entire acquisition, 1.8 second acquisition, against just using only the 0.45 acquisition. Right? So I'm just using the front end of the same signal, only the first 1/4, I believe, yeah, instead of the whole thing. Yeah? Here is I just introduced the integral based answer as well. So the blue line is the integral frequency domain calculation of the amplitude for, I called it 16 through 0.125. One is 50 to 1 signal to noise, right? Our conventional signal to noise of peak height to noise measurement. So I just use that as my standard. 50 to 1 is standard. Anything below is smaller. So this is 6.25 to 1 in the conventional signal to noise measurement. You do an FT, measure the peak height, measure the RMS, calculate your signal to noise. 16 would be significantly higher, 800 to 1 signal to noise. Yeah? So that's what the 16 to 1.125 mean. So the blue line is the accuracy of your integral of the values. Yeah? I'm expecting 100 because it's a synthetic signal. I put in a value of 100 in my amplitude. Right? That's your integral. Why does it go down when the noise level goes up? I don't have to talk about that. Go read Gennady has published a nice paper recently, last year, in analytical chemistry, discussing how the noise affects integral calculation. Using some Monte Carlo methodology, computations and things like that. It's a beautiful paper. Just go and read that. I'm not going to talk about that. Here it's not a comparison of frequency domain to time domain. I'm talking about comparison of time domain analysis to time domain analysis. Yeah? So the yellow is also frequency domain, but it's a modeling approach. The red one is time domain analysis using the entire FED, 1.8 second of the FED. The orange is the same FID supplied, but I said use only first 25% of the FED. Don't use the entire FID, just use first 0.25% of the FID. And then that's your error value. So this is the value that it calculates, that's the error, what it's supposed to be, what it is not. If you're looking at, that's a 1.5% error from what I'm expecting with a signal, with the data that has a signal to noise, conventional signal to noise of 6.25 to 1. I think if you are doing quantitative NMR, and if I give you a data that has a 6.25 to 1 signal to noise and say, go give me a 1.5% error in there, you'll tell me I'm crazy, right? I don't have to tell this group about quantitative NMR. I'm probably the novice among all of you about quantitative NMR, but that's pretty impressive. Now, signal to noise is a measurement of uncertainty. The uncertainty can be how poorly it detects, meaning the accuracy of that, or if I repeat it multiple times, how good are you able to reproduce every time? So here is the data that I did. I just took generator synthetic signal and the noise of a singlet, right? Different noise values, all of them about signal noise about 11.3 plus minus 0.7. So I have about 11 fifths like that, right? I analyze 11 fifths. I want to do the same thing in all 11 of them. Noise is random, the same signal. Yeah? See how can you reproduce. So this is the value there, and so this is what I'm expecting because I introduced that signal. Yeah, this is my signal. There's the amplitude reference, and integral gives you that, a lower value, but it has a spread. That's the craft results or a time domain analysis result using the entire FID. The value is very close to accurate that I just showed you before in the previous slide. There is a spread. But the moment you start to use only very early part of the FID, I don't care about the rest of the FID because I don't need to separate my signal. It's a singler, for God's sake. Why do I need more than certain number of points in there, right? Look at the tightness of that error. So the reproducibility is also better. That means the measurement, uncertainty in the measurement is better if in the early part of the FID. In time domain, signal to noise is a different definition, is how you analyze the data. Which part of the FID you're going to analyze the data with. So shorter acquisition time decrease your uncertainty. Counterintuitive, but it is. Here is an example of that. So this is actually a reaction monitoring using F19. Yeah, that's trace number 1, trace number 50, trace number 100, meaning they had some kind of a 1 minute kinetic time point. So this is 50 minutes later, that's 100 minutes later. There are other FIDs in between I'm not showing you. And they want to monitor that particular peak. So here is an expansion of that peak, real peak. Yeah. So if I do a time domain analysis of this across the timeline, kinetic timeline, that's the data I get. Looks pretty decent, if you may say, for the noise that it had. Yeah. But I'm using the entire FID. So I simply said, nope, I'm not interested because that F19 is well separated from the rest of the guys. I have no problem in frequency separation. Just use 0.05 millisecond of that same FID, the same input. I just simply said, just use the first 50 milliseconds. Don't use anything else. Signal to noise there is very high in that FID. Uncertainty between transient to transient to transient, if you want to call it, is better, much better. Much less uncertainty in there. Uncertainty variations in there. Yeah. how do I increase my signal to noise in a conventional fashion? This is a very common practice, yeah? Dates back to invention of FT-NMR, yeah? Here is a single scan, assume there's a single scan FID, signal to noise of 100 to 1. Let's talk it. So if you want to increase, time average. I do a second scan, signal increases by a factor of 2, noise increases by a square root of 2, signal noise increases square root of 2. So third scan, fourth scan, and so on and so forth. What if I have a probe and a hardware that gives me a signal, NMR signal, noiseless? Violate all fundamental laws of physics and electronics. Let's, for argument's sake, I have a probe and a spectrometer that gives me no noise signal. And if I can add that signal to this signal, 100 to 1 becomes 200 to 1 because I'm not adding noise. I'm just adding signal. But that signal need not necessarily be a physical signal. What if I can describe that signal as an equation? If that signal, noiseless signal, is an equation, I can add that equation. Equation is an exponential decay function. It has a frequency, it has an amplitude, it has a decay rate, it has a phase. If I can add that to the same FID, yeah, I can. In other words, I am essentially spiking that FID with a model. Right? If I add that, I don't have to. I am not limited to adding 1 to 1. I can add 1 to 100, 1 to 200. So I can increase my signal noise of that FID 100 plus 100 is the original one that I had plus n, my multiplication factor of that frequency, of that equation, and add it to that. I just whoppingly increased my signal noise n times, yeah? I can do that because it's just an equation. And I say add, I just say multiply that by 100 and then add. So the key point is I must have an equation to represent that signal. If I can have an equation to represent that signal, I can do exactly that. This is exactly what you can do too. So let's see if I can do that. That's a reaction in a THF. Right? So there's a reactant signal. This is THF. That's an intermediate signal, some intermediate compound that is forming. I have no idea what it is. I'm just interested in some data. That's a product. Right? As your kinetic time changes, the reactant goes away, intermediate forms, and the product comes up. So if you look at it from time domain, this is a time domain analysis. Your reactant goes down, your intermediate goes up and down, and your product comes up. But where is the most interesting part from a kinetics point of view? Most interesting, important part of your kinetic study. It is in these transition positions. That's where your kinetic properties are buried in. That's how your calculations are going to be. It's useless, really, at this point. But this is where my uncertainty is lower, because my signal to noise there is very high. So my uncertainty from transition to transient to transient is lower there, whereas here is where I have the worst thing. Where the most important part of my kinetics, is where I have the worst problem, because my signal is just growing or my signal is just decaying out. How do I improve that? It's very simple to do, because if I just look at this FID and then say, okay, the trace number 3, I don't take trace number 1 because maybe the reaction hasn't started, they haven't fired it up. I take the trace number 3 and then say model that peak. And then I take trace number 25 and then say model that peak. I take trace number 111 and then say model that peak. So now I have those 3 models of a reactant, the intermediate, and the product as equations. Those models are synthetic models, meaning I just fit it to an equation. I have an exponential decay function. I can simply add it to all the traces and then say, go do the same analysis. I can just add 5 times that signal. Look at that data. The same data. All I did was just, instead of analyzing from trace number 1 to trace number 111, I simply said, analyze 3, 25, and 111, take those model and add them all to everybody. And then redo the analysis for the whole thing. I just increase my signal to noise by a factor of 5. So my uncertainty is much less. So my shapes of my kinetic curves are much nicer. I can probably trust that much better than something that's going jagged edge. Just to show you that it is really the real data, I just didn't present a function. That's the Kraft result. It's 5 times more signal than what it was. That's the Kraft result. But if I want to be very nice to look at, I want to look at it properly. I don't like what you are showing. I don't know whether you're cheating me or not. Because I just added this model. I can subtract the same model from the final fit. If I subtract that, that's a Kraft result. Visual result. Yeah. So it is exactly same as your input FT result, you get the Kraft results. But this is the interesting thing. All of a sudden, by doing nothing, no addition of data, no collection of data, I just increase my signal noise because I have an example model. I can do that. Here is another thing that you could do. What if we had examples here that we heard about, I have a very tiny material. I want a very small signal I want to quantify. How do I do that? I need to increase my signal to noise. Whether it's benchtop or high resolution NMR, whatever. When your signal is very small, you just want to somehow increase the signal to noise better and better. What if it is that peak is what is interesting? So this is a trace that at a vertical scale plotted in the normal fashion. That's the same data just plotted with a blown up vertical scale. And the peak of interest is this shoulder. I want to improve my ability to quantify that signal better. Don't ask me to go back to the spectrometer. That's the only rule. I cannot go back to the spectrometer. How do I do that? Very simple. So this is just to show that that's actually a real signal. If I have a reference standard, many times in our lab we have a reference standard. I have a reference standard. So here is a reference standard material that's what I am trying to quantify. If I know that, I simply say, okay, give me the model of that reference standard. I just want to generate that, convert that into an equation. That peak, I want it to be as an equation. Once I have that peak as an equation, I can add that model into that big trace on the top, to the FID on the top. I don't have to just add 1 to 1 or 2 to 1. I can now do a titration. I can say add 0.8, 0.85, 0.9, 0.1, 1.1, 1.2. I can just keep adding, titrate, and not just take one value, take 11 values, plot a 1-to-1 linear plot, because I know exactly what I added. I look at the amplitude and then plot it. The intercept is the quantity that was originally there. It's a basic linear equation. And you can do a digital equation, digital titration, and there is a peak which says that it is 0.024217 with a standard deviation from the linearity that I expected because I asked it to do multiple Kraft analysis using the same model, using the only fit that I had in my hand. Let me show you how small that is. This particular trace is done 1x vertical scale. That is 10,000x. And that's the peak we are looking for, without going back to the spectrometer, without recollecting data. All I had was, I had a standard. This is very, very common in almost all laboratory practices. When you don't know anything and you want to quantify something, you have a guess of what it is. Or you are looking for an API, or you are looking for an impurity. This is a very common scenario. And you have an opportunity to do that. You can do that because you are doing it in time domain, because you convert that into an equation, and you can equation that is a noiseless FID. Suddenly you have an opportunity to change your signal to noise. Something very basic. I did not do anything magic other than just convert that FID into an equation. That's it. It gives me opportunities to do things that I never thought I can do. Resolution is the second property that we are talking about, sensitivity resolution, is yet another confusion. Everybody talks about resolution in their own terms. This is a definition galore in literature. I'm not going to define one or the other. I'm just pointing out there is a definition, multiple definition of what we mean by resolution. I can include more terms to that resolution, the termed resolution. I can say resolution is ability to rip off your FID, your spectrum into multiple components. That's a resolution. Or something like this on this side, right? That's a resolution. Here is an example. Completely different definition of resolution. This is an antibody spectrum. And that is a spectrum from the same FID, not going back to the spectrometer, not a CPMG experiment. From the same FID, just show me only those models that has line width somewhere between 30 hertz and 6 hertz. Why did I pick 6 hertz and 30 hertz? I don't know. I just said, okay, let's pick two model widths and then filter only those models out. And that's a spectrum. I resolved a medium size, if you extend this argument, I resolve a medium size molecule or medium line width models out of this mess. Just because I am able to do a time domain analysis, meaning just because I am able to convert my FID into a set of equations. And I can filter that. I can also filter peaks that are very narrow, sharp peaks, impurity in small molecules in that same mixture glob. I just used the antibody spectrum as an example. It can be anything. It can be polymers. It can be monomers in your polymer. It can be a polymeric material you are synthesizing from glucose. I don't know. This is the kind of stuff that you could do. Just because I converted the FID into a set of equations. And the moment I have a set of equations, it's a spreadsheet. I can do all kinds of things that a spreadsheet sorting mechanism do. I can do it with this. It's a spreadsheet, Excel spreadsheet, nothing more. Resolution becomes an interesting concept here. This is a resolution too. I have a doublet. This is true example. This is a quercetin data that I got from Guido Poli's lab. It's a real data. It's a 2 hertz line with 2 hertz coupling constant resolution, right? So these are the 3 doublets that we were analyzing. I could ask them to go back and then generate me data that is broad data. So all I did was I just took that FID and then just added a line width to it. I multiply by some exponential function, right? So this is equivalent to saying that that's a spectrum of quercetin with a line width of 5 hertz because you didn't shim, right? And can I get 2 hertz line width out of 2 hertz resolution out of that? Because my coupling constant is 2 hertz. The answer is of course yes. It doesn't matter which FID I take. The FID that has a 5 hertz decay, very fast decay, or the FID that had a very slow decay. I can take that, and here are the, within certain limits, of course. These are all the same coupling constants. And that's your in a spectral display. So the first one, I'm going to go back again. The first one is it's as it was, and now it is 2 hertz, a line width added, 4 hertz line width added, 5 hertz line width added. So the blue line is basic FT. And the 2 top lines are what it said. That I can fit that into 2 models of 5 hertz each. They are separated by 2 hertz. They are separated by less than the 9 bordering function that I applied. And it says that's the best way I can fit. And this happens in every, it doesn't matter. So frequency separation permitting is a term that I put in before in one of the definition of signal to noise. You just use only front end of the FID. Signal to noise separation permitting, you can do that. Here, where is the separation permitting? The decay of the FID is 5 Hertz decay. And I have 2 Hertz separation. You can separate that. Because it is a modeling tool. It's not just a mathematical Fourier transform. It's a modeling tool. It says, this is the best I can model. And it gives me, there is no precondition I apply. I don't say, I want you to separate 2 Hertz separation. I simply say, just rip it apart and give me all the equations that will fit this FID the best. And it comes back and then says, that's how I can analyze. So this is very common in all 2Ds that we collect, meaning all of our interferograms are truncated. We collect for many, many times 64 increments in carbon dimension. Think about your acquisition time in that dimension for 64 points. It's in less than about 2 milliseconds at the most you collect. And then you want to do the separation, sure, you can do that too. So this is a truncated FID. If I have this, so this is your input FID, output meaning Kraft output, that's a residual. If you do Fourier transform, you get something like that. But the interesting thing is, this guy is not just a simulation. It's not an output of that analysis. It is a simulation of the output, because it is simulation of that equation. Nothing tells me to stop at this point in the simulation. I can essentially simulate it as long as I want. Right? If I now Fourier transform, look at the sharp line there. Right? You can exploit that even better. This is a 2D analysis of something wood sap 309 increment on a 700 megahertz spectrometer. It's a wood sap extract, 700 megahertz spectrometer, 309 increment. The orange line that you see is a conventional Fourier transform results. It's a mess. That's what it's expected to be. It's just a wood sap extracted. Right? And the blue ones that you're seeing here are Kraft analysis simulated to present it to you. It's a table represented as a 2D. The bottom trace is actually a depth quality 1D spectrum, carbon, from the 309 increment data. That is, it's not a projection, it is a sum. Compare that to the orange line there. That is a projection of the 2D the way we have been doing so many years. It is 2D transform, project. Let's blow the vertical scale up. That's a 2D transform project. The orange line behind is a 2D transform projection. The blue line in the front is actually a full-fledged simulation of the table. Nothing is new added, nothing was taken out. I just said, here is a table, sum them all up and give me a fit. And the resolution is fantastic. Here is something else you can do, because it's a spreadsheet, right? Here is what I'm doing. I simply said, take that projected or summed depth result, take only peaks that are less than 25 hertz wide, because it's a wood sap, remember? It has large, broad resonances as well. Take only the narrow ones, and I want you to show me only peaks that are less than 5% of the biggest peak there. In other words, I want to look at this impurity. I want to look at this hash in there. I don't care about the rest. All you're doing is you're taking the spreadsheet and then said, anything below this and anything in between that. Take a table, generate a subtable, simulate. And that's what that is doing. So this is your full spectrum. That's my filtered spectrum. All of a sudden, I am doing a filtered experiments with no spectrometer. This is a filtered experiment that we can't even think about. It's a filtered experiment of peaks that are narrower than certain window that you can supply any times, any number of times, and only peaks that are smaller than certain value. And you can supply any times, any number of times to that table. It's computational world. No time expense of your spectrometer. It's a CPU time, period. And you can, I have a lot more data on this one. You can look at this. It's like a multi-dimensional Rubik's Cube. You look at it in whichever way you want. You rip it apart and look at it in a different way you want. Now you have an intense amount of information. Intense amount of information from 309 increment HSQC on a wood sap on a 700 megahertz spectrometer. One data collection. This is what you can do. I haven't done anything differently other than just looked at the basics and then say, the moment I get it into tabular domain, I'm just applying the same basic principles. Here is an HSQC of a sugar, anomeric region. Classic 64 increments, 16 scans per increment is actually a quantitative HSQC. And these are your acquisition time. And the red lines are the standard Fourier transform result, and the blue ones are the Kraft results. And here are separations that you can see. Here is 3 separations you can see. 64 increments. And those separations in the carbon dimensions are 3 hertz from one another. 3 hertz, those carbons are separated. And the interesting thing is some of these are separated in 3 hertz, not only in carbon, but also in proton. They are even on a 2D, they are cross peaks that are that close to each other. I'm going to pause that one and come back after a few minutes. How am I doing in time? Okay. Okay. I am going to hold that door. One more, last one. Line shape. Basic line shape. Yeah. We talk about, that's a fun basics. That's a basic. Here is 2 spectra of the same sample. Look at the line shape, look at the line width at half height. Exactly the same. Maybe 1/10, 1/100th of a hertz off from each other. But whereas the line width at 0.55, which is measured on this, I think on this line, it's measured on, this is a measured number. This is 20 hertz line width, that's 10 hertz line width. Under any practical NMR guy's point of view, the blue spectrum is the best spectrum. No questions about it out of the 2, right? No questions. Nobody will question this. You can actually put one on top of the other and you can see this. Obviously you can see that the blue is narrower, blue is taller. In the previous slide that I showed you, they are plotted in the same vertical scaling factor and blah, 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 right? So what about time domain analysis? What does it do? It analyzes either one exactly the same. So this is your FT result. That's the sum of all the models. That's a residual. Both of them are ripped apart properly, completely, right? No questions about it. I have actually a slide to blow the vertical scale. You trust me that they are all very well done. I'm putting this statement here. It's important to, the goal is not to define a resonance with a single best model. It's not like fit that singlet to a model. I simply say fit this signal to as many models that you need to completely define. That's a rule in this game, right? It's not about, I don't precondition that and then say, that's a singlet, these guys are doublets. No, nothing. I have no idea what this fit is. Take as many exponential decay function as you want to fit that completely. That's the deal, right? Here is a rip apart of one of the fits. This looks like Every resonance is one model. That's a rip apart of the other fit. All it did was it said, there, it's not Lorentzian, it's not a good, I have to use more models in order to define each one of them. I have to use more models to define each one of them. The complex sum of all these models gives you the input FID that you, fit the input FID that you supplied, period. Yeah? So that's why your residual is 0 in either case. So both of them got ripped apart properly, defined properly. We know one is a good fit, another one is a bad fit. The blue fit is a good fit, the red one is a bad one. Except it's the opposite. It is a red one that we thought is a bad FID that ripped apart properly. The blue one, which we thought is a good FID, rip apart is multiple. It needed more models to define that. Why? The one thing that I did not tell you is Lorentzian equation. Fundamental, 0.55 is approximately 13.5 times width at the half height. Your half height is 1.38 hertz, multiplied by 13.5. If you do that, it will become to around 18 point something. In other words, the blue spectrum, sorry, the red spectrum, actually the peaks are closer to the Lorentzian. So that is better shimmed. If you believe NMR signals are Lorentzian or NMR signals are exponential decay function, that's your endpoint of line shape should be. Not the best one that gave me a highest peak. That's simply because we all lived in the frequency domain area. The moment you say, my NMR is exponential decay function or Lorentzian, that's what you should be shimming for. That's what you should be shimming for. It's not. So I'm going to skip this slide and then ask the fundamental question. So what? So what's the big deal? You just gave us a BS about Lorentzian, non-Lorentzian. Your craft ripped it apart anyway, perfectly fine. So what's it? The problem is this. Signal analysis, facts of life. All data analysis involves comparison of to a model of a data. There is no substitute for good signal to noise. Signal to noise that I just defined on the very front of this lecture about signal to noise is different in time domain versus signal to noise in frequency domain. Now it all congeals together. Yeah, these are all important. So if I take something like this, a singlet, and then I say rip it apart, if the goal is, I put this mantra about goal is not to define a resonance with a single best model, but to define as completely as you can with as many models as is needed. Yeah, if you do that. But what caveat is, given adequate signal to noise. Meaning, if this minor model that it needs to correct for the non-Lorentzian line shape of your F signal is very low in signal to noise, it's going to say, I have too much uncertainty there. So signal to noise is important, but line shape is important as well. Both are important. So that we can, so this is my mantra in a way. Attempt or limitation to fit a non-Lorentzian singlet. We are all doing this in the world now. Any modeling tool that we see is that Lorentzian? No, but I'm going to fit this. Is it Gaussian? No, I don't know. I'm going to fit it to Gaussian windows. But the fundamental, this is not about NMR. The fundamental truth, this is the moment, is anything that you try to fit to an equation should be a linear combination of that equation. It can be a financial analysis in the Dow Jones Industrial Average. I don't care. If you're trying weather prediction, I don't care. If you're trying to fit it to a model, set of models, that has to fit into that set of model. If not, you are compromising. I'm sorry. you're not getting the real result. You're compromising because you don't know. But now we know. We know NMR is an exponential decay function. NMR is a Lorentzian line shape. It's a first order kinetics. NMR signals decays first order. Other than some spin interactions. I won't go into that. So, actually I did those things by fiddle or not to fiddle, that's all. I asked a question, so what? You can actually take a non-Lorentzian signal and convert it to Lorentzian using the tool called fiddle, that was published by Gareth Morris. That's a slide I just skipped because I am running out of time. So what? Here is an example. This is 3 carbon-13 spectrum of sugars. The same sample, master sample in 3 aliquots, and they ran 3 spectra. Yeah? So all 3 should give you the same data. Obviously, from an analytical spectroscopy point of view, there's going to be measurement from all 3, and then finding out what is the variation is an important criteria. Reproducibility, standard deviation, standard statistical analysis is important. There are peaks that are here that are about 3 hertz away. This is the sugar that I alluded to before. But it is poor signal to noise. It's a quantitative carbon spectrum, poor signal to noise, because their delay times are 200 seconds. D1 is 200 seconds. So they don't have enough sample to run that way. That's a poor signal to noise in the conventional fashion. So I can immediately say, aha, time domain, focus on the front end of the FID. Yeah? I can do that because the peaks are separated. There are peaks that are separated by 3 to 4 hertz. So I'm limited because I said good signal to noise in the front end of the FID, frequency separation permitting. Here frequency separation is not permitting. That's 2 or 3 hertz from each other. So I have to use a certain minimum amount of FID. I can't just use first 10 points. If it's a singular, you don't even need more than a point, yeah, if you're quantitating. The first point is your signal. So I am limited to the tricks that I had in my mind. Yeah. The last one is highly likely they are all non-Lorentzian. I'm going to bet my nickel on this because we all shim it very well. That's why they are non-Lorentzian. We shim it to 0.6 over 6 over 12. Guess what? 0.6 to 6 over 12 is not Lorentzian. Just go do your calculation. They are non-Lorentzian. But that's a spec. So I shimmed it to my spec. Yeah? So highly likely these guys are not. They are all non-Lorentzian line shape. So how do I solve this? This is when the fiddle comes into picture. The fiddle, or taking your FID and then converting back into a Lorentzian type of line shape, is very useful because if I don't fiddle, the red bars are my standard errors, standard deviations from among the 3. I have only 3 points. I agree, but it's just 3 points. Red, blue is if I just do a fiddle and then do it. So by converting them into a Lorentzian line shape, I could still use the same rules that I have. I can do better, right? This is the truth. This is actually the plot of an expected concentration of these sugars against the concentration of the sugars from carbon spectrum plotted against each other, right? That's a 99 R squared value with those kind of signal to noise. 10 to 1 to 45 to 1 is a range of the signals there. Now get back to the HSQC. Why do I not do HSQC? Why do I run carbon? I can plot the HSQC result against carbon. That's again 0.98 correlation coefficient. So because the previous slide I had showed you, expected concentration to carbon is 99, carbon to HSQC is 0.98. You can say expected concentration of HSQC is 9. This is still a quantitative HSQC. Long relaxation for proton, you have to relax, and you have all these pulses that you need to properly apply. You need to have these strict pulses, some nice shapes that comes out of in literature. This is a QHSQC. My question is, fundamentally, I just said a few minutes ago, a lot of people, we have a known compound in our hand. We know many times. I'm not saying all the time. Many times, when you're doing a quantitative run, you have a pure compound. You have access to a pure or relatively pure compound that you are trying to quantify in a mixture. Many times in those scenarios, why do you need to do quantitative HSQC? To me, it's waste of time. Because we all trust our NMR. If I run scan number 1 and scan number 2 within the same 2D, I am supposed to get exactly the same thing. That's why I'm able to time average properly. We all believe in that. We do this all the time. So if I run an HSQC of a compound first and immediately follow HSQC of a mixture, you can probably assume the same argument, assume they are all the same. They will behave the same. The same compound, we run number 1, run number 2 will behave the same. If you run the same data, same way. If you can process the same way. In tabular domain, you can do that. So here is an API. This is a standard Q1 HSQC. This is nothing to do with quantitative HSQC. It's just a bloody crisis HSQC, nothing else. HSQC spectrum run. Standard API, I call it API. It's a mixture. Here is a table. Here is a table that came out of the craft results. I can compare. I can get quantitative with a plot of concentration. If I know this is 20 millimolar, I know that it's 5 millimolar. We do this all the time in proton spectrum. All the time. We have this Kenomics, somebody called Kenomics software. They have a bunch of data that we collected at specific concentrations. You compare that against them in a metabolomics mixture and then say, my concentration of acetate is 2.32 millimolar because I'm comparing against somebody's database which has a 10 millimolar acetate spectrum. We do this all the time. I'm just saying, why can't we do this in 2D? What's the difference? What's the difference? You could do that in 2D. So you can compare that. You can generate your API spectrum, generate your API quantization. You can generate your impurity spectrum. It's just a subtraction. All tabular. Nothing to do with a spectrum. Spectrum is visualization. Spectrum is nothing but visualization. I can do this even more fancier. What if I do, oh my God, I did my API with normal HSQC, uniformly sample. I have this mixture. I'm running 50% non-uniformly sample. How can I compare? Of course you can compare. What does that got to do with uniformly sample? How do you sample your fit? Your amplitude is in point number 1. Amplitude is magnetization at time 0. That's quantitative NMR. So how you collected non-uniform sample, uniformly sample is all how to separate that, not what it is. So here is an example of 5 millimolar, 20 concentration difference, number of increments different, non-uniform sampling, density difference, number of scans per increment difference. You can plot against the reference any which way you want. They are all greater than 0.99 R squared. You know what? It simply tells me the spectrometer works perfectly fine. That's it. I didn't do anything. I just trusted the spectrometer. So the spectrometer works, reproduces what I say it should reproduce. So last slide, I'm done. We all know ASAP HMQC, yeah? Very nice, very clever way to speed up your 2D spectrum. I just said you don't have to do a quantitative HSQC if you want to do a quantitative NMR. If you are doing a relative quantitative NMR, you don't have to do it. I just said that. And I showed you a whole bunch of results. Let us take an ASAP HMQC. Invariably, you can run it in 1 minute. Because these things are. Every increment, you just do that. Right? So that means it's 1 minute acquisition, maybe a minute of processing, because you are not doing the conventional Fourier transform. You're doing something silly called craft or something else. So 1 minute process, every 2 minutes. Every 2 minutes, it spits out a table. What table does it spits out? It spits out a table of carbon chemical shift and an amplitude. That's carbon chemical shift. It has 2 carbons that are separated by 0.2 hertz, 0.2 ppm. If I want to, one is a product, another one is a reactant, I can follow that. That's just 2 separate lines in my table. 1 minute acquisition, 1 minute process. Why can't I do time course monitoring of C13 NMR and follow that amplitude? So I haven't asked you for anything other than what I just said. Given all that what I said before, that till the previous slide, why can't you do that? C13, natural abundance C13 quantitative reaction monitoring, 1 second, 1 minute time course interval. I see no reason why you cannot do that. The only reason I see you cannot do that is you just don't want to do that because it's something different than what I am used to. I'm just saying it's not. You can do this on benchtop, for example. Here is a benchtop spectrum, 128 increment benchtop spectrum, 64 increment benchtop spectrum. Kraft result is exactly same. In Kraft, the spectrum is visualization, nothing more. It's exactly the same. I can plot the amplitude of this against amplitude of that. I get a 0.95 correlation coefficient. I'm sorry. That's pretty good. That's pretty good. On a benchtop, 64 increment. 64 increment, I believe, probably 8 scans. For a benchtop, that's good. Why can't I do reaction monitoring, benchtop, HSQC? And if many benchtop these days come up with gradients, why can't I do an ASAP HMQC on a benchtop, reaction monitor in a factory at 1 minute kinetic interval? Why can't I? We have to ask that question. Because in order to move forward, we got to let go. With that, I stop. Oh, I still have to. I still hold the mic, right? Any questions? Drinking, yes. Me too. Thank you. Oh, any questions now? That's fine. Any questions now? That's fine. Yeah, Krish. Is Krish against Krish? Okay.
Thank you, Krish, for the excellent talk. So in your antibody spectrum, you showed us a great example, and you're able to filter peaks based on a given line width. Yeah. So what would you define number of model necessary to reproduce the actual spectrum of that antibody?
One of the fundamental rule that we supply when we do craft is we don't say how many models it needs. We simply say keep digging in until the residual is random. So there's no rule. How many models to fit that is no rule. It's an iterative process. So imagine the Bayesian works this way. The first question to Bayesian is iteration 1. Given no precondition, is there a probability of finding a sinusoid? It says yes, there is a good probability, then find me the best sinusoid that will fit. So now I have a potential model that will fit that, but there may be more. Right? So then the second iteration, given this, there is a potential model there. What is the probability of finding another model? No other condition. What is the probability? That means is the leftover point completely random, or is there something sinusoidal sitting there? And then it says yes, there is. Then what we say is take both the models, iterate their amplitude frequency to best fit, and now you have 2 models that best fit the given FID. And then we say, given these 2 models, take that out first, is there another model that's potentially possible? Then Bayesian will say yes or no. and then you iterate. So you keep iterating until Bayesian comes back and say, no, the leftover, after given the n number of models, that n is not determined by the user, but by the Bayesian itself. After having n number of models, is there another model, potential probability of a model, the Bayesian will say, the leftover randomness exceeds a probability of finding a sinusoid. That's when it stops.
Sorry, the question then is that doesn't you were at the end of the model already coming down to exactly the spectrometer of noise?
Exactly. Exactly. So that's why in the mantras, one of the first one is there is no alternative to true signal to noise. Decrease your noise, there is a value to that. No question about that. There is a value to that. In fact, there are other ways we can increase the probability of finding. I think about 3 or 4 years ago, I talked about this. Oh, am I okay? Okay, 3 or 4 years ago, I talked about this. It's actually not collect the data longer or no more number of scans, but actually oversample. If you oversample your data, in other words, you keep your dwell time 4 times, 1/4, or spectral width, blow your spectral width out. From a frequency domain point of view, it's useless. From time domain point of view, it's very, very valuable because you take the same sinusoid, now you defined it with more number of points. So the probability of finding the smaller and smaller models is higher because you have more number of points where the signal to noise is maximum, right? High, right? So oversampling a FID, which I didn't bring it up today because I didn't think it was basic principle, but oversampling is incredibly useful. We do this all the time in craft, oversampling. In fact, we have actually that paper that I published actually have a trick to oversample a normally acquired data. You can do a Fourier transform, inverse Fourier transform type of way. You can oversample, but oversampling certainly improves your dynamic range. I want to call it a dynamic range here, right? Because Bayesian is going to fit based upon the most probable model, invariably the most abundant model or the most least slow decaying model. Yeah? Those are the 2 criteria. Probably will fit that condition. So it's going to go from there to the least amplitude. Oh, sorry, I keep, okay. So it's going to go from the most abundant to least abundant and long lasting to fast decaying, right? In that combination, right? So having a high signal to noise certainly helps, but signal to noise doesn't mean 2 seconds. If you can live with half a second, collect more data. It doesn't cost you anything, right? Because we oversample all. Every spectrometer oversamples. I can bet you all spectrometers oversample data when they collect. All spectrometers, including benchtop. You know what we do? We just immediately shoot ourselves in our foot. We say, I'm interested only in 16 ppm. Don't bother to show me the rest. Downsample it right there. Waste our time downsampling it. That oversample FID has so much information that we just shot ourselves in our foot. We do this all the time. Unfortunate, but we do. Except I think Magritech doesn't. Magritech keep it 100 ppm, proton spectrum. Very clever. Very clever. Thank you.
So thank you, Krish. And I think we'll move on. I'll give the mic to John for some last words.
So let the record show I'm not holding. John is holding the rest of them from the reception.
Yeah, the bar is open, and I'll not take more than 45, 50 minutes. Kidding. Thank you very much to all of our speakers. Absolutely fantastic talks. Worked out great today. Is work out good for you guys? Worth your while, I hope. And got ENC starting tomorrow, of course, and MR Resources Q1 and Ivan will be right here in Oak Shelter. Join us we'll be here Sunday, Monday, Tuesday, Wednesday, but in particular, join us all 3 nights, Monday, Tuesday, Wednesday, if you can, for our now, dare I say, famous Ivan hours that we're doing. Fun, quasi-NMR-related talks, that is, talks given by NMR experts, but not necessarily about NMR. We've got one night on, Ron Crouch is going to update us on the world of astronomy. Another night, you old-timers are going to especially like this. Look back at the old Texas A&M University NMR newsletter. And one of the other talks, I don't remember which night is which, but will be a look into the world of fine art via NMR. So that should be pretty cool. But once again, thank you everyone for joining us today. Dave, any final words?
I guess my own final words are thanks for being here, and I enjoyed it, and let's head for the bar.